Good morning or good afternoon, depending on where you live. On behalf of the Canadian Honey Council, I want to welcome you to this educational webinar on Varroa control. The Canadian beekeeping industry has been experiencing higher and higher levels of overwintering losses, and the Canadian Association of Professional Apiculturalists, in their overwintering report, has identified Varroa as the primary cause. Today, you're about to hear experts in the field providing information on management skills to improve Varroa control. Beekeepers across Canada are aware of the decreasing efficacy levels of various Varroa treatments and the need to add more tools to the toolkit. The Canadian Honey Council, researchers, and companies such as Véto-pharma are working to achieve that goal. But in the meantime, we must deal with what we have to the maximum efficiency. The CHC is especially proud to help sponsor this event to assist beekeepers in maximizing their efforts in Varroa control. And it's important to note that there will be other webinars later on in the year that will also be discussing this topic. I would like to thank the tech transfer teams from across Canada for providing content on this program. Their valuable work with beekeepers continues to be a beacon of success in battling Varroa. But all this could not be occurring without the support of Véto-pharma, and in particular, Miguel Rodriguez, the bee business director, who initiated this project, and Enrique Marsque, and the team who were able to pull this off in a very short timeline. It is truly their work and support that made this possible. So sit back and enjoy, and hopefully take back valuable information to improve your operation in combating Varroa.
Well, first of all, thank you, Rod, and welcome everyone. Thank you all for your interest in today's webinar, Staying Ahead of Varroa, Challenges and Strategies. My name is Ulrike Marsky, and I am the Technical Manager at Véto-pharma. I'm joined here today by two of my colleagues, our Managing Director, Miguel Rodriguez, and our Marketing and Communication Manager, Joanna Collin. And in the name of Véto-pharma, we would first like to thank the Canadian Honey Council, as well as the tech transfer teams for apiculture, for taking time out of their very busy schedules to organize this webinar for beekeepers. We're very happy with the program, which, as Rod has already indicated, has been prepared in a relatively short period of time. And I really hope that everyone who is following the seminar today will benefit from the information presented here about bee health, Varroa management, resistance management, and related topics. We would also like to thank the presenters of this webinar today for their time and resources in preparing their talks. Phil Lester from New Zealand, Geoff Wilson from Saskatchewan, who will be joining us today from Thailand, Julie Ferland from Quebec. And in the panel discussion later on, we will hear Andrew Byers, Matthew Polinsky, Medhat Nasr, and Maggie Lamothe Boudreau. We will introduce all of the speakers in a little more detail later on, and all of the panelists as well, right before their respective talks or discussion. I would like to stress again that we really hope to turn this webinar into a series that will continue with additional topics revolving around honeybee health later this year in autumn or in early winter. Thank you. Let me introduce our first speaker, Professor Phil Lester. He is a researcher at Victoria University in Wellington, New Zealand. And the key focus of his research involves the development of safe and effective control options for pest species, such as Varroa mites. He works on next-generation approaches that include immunotherapy and the potential of double-stranded RNA for highly targeted pest control. Phil's talk for today's webinar will be about integrated resistance management for acaricide use on Varroa destructor. And after the talk, we will have a 10-minute session for questions. And then after that, we will have a quick 5-minute break before the next talk. So I will hand over to Phil.
Thank you very much. Hopefully you are able to now see my screen, and that's all good, and sound is coming through well. Cool.
Perfect.
Thank you, and thank you to Ulrike, Joanna, and Rod, and the team for the invitation to speak today, and to the Véto-pharma and Canadian Honey Council. It's my pleasure to be here from New Zealand, although it's a little bit early in the morning. Right now it's about 4:45 and 5 o'clock in the morning. As I already suggested, I'm going to be talking today about integrated resistance management for acaricide use in Varroa destructor. What I'm going to do is go through really 4 bullet points effectively in the next 40 minutes or so. Talking about Varroa as a super pest. Lots of you will be aware of Varroa. I'll give you a little bit of rundown anyway about it, and what pesticides and integrated resistance management actually is. And it's really important for the efficacy and longevity of our synthetic chemicals and their use in this particular pest. I'm going to go on over 2 problems with Varroa in terms of integrated resistance management. It's a particularly challenging pest to manage, unfortunately. Then we're going to talk about some methods and attempts for resistance management in this population. And I will conclude that actually we need to do a lot better than what we have been doing in terms of integrated resistance management with this particular pest species. So let me give you a little bit of a rundown. All of you will be very familiar, I am sure, with Varroa as a super pest, really. It's a super pest. Super pests are defined in the literature a little bit as a species with a global distribution, and now it does have a global distribution now that it's come into Australia and established a strong foothold and is spreading throughout Australia. It's a pest since the 1950s, as you'll all be aware. And here in New Zealand, we first identified it in only 2000, so only 25 years ago. It's probably present 3 to 5 years prior to that. It probably came into Auckland, somewhere up around here in New Zealand, and then spread through the country. Probably just one introduction. Probably we think a beekeeper probably had a pocket of bees brought in from overseas, and some bees in his pocket, and they had mites on them and spread throughout the country. New Zealand has deformed wing virus A, but not deformed wing virus B. And interestingly, Varroa in Australia doesn't have any deformed wing virus at all, it seems. So it's managed to invade there without it whatsoever. It's these viruses that are often the big effect, as you'll all be aware. Here in New Zealand, it's increasingly a problem, like it is everywhere. So this is some data going back from 2015 up to 2023. And it's showing the overall loss rates, winter loss rates of Varroa in New Zealand. And we started off in 2015 about 8.4%. And it's gradually climbed over that time. And now if we include both winter and autumn loss rates, we're up to around 17%. So it's by some standards, including those in North America this year, it's not necessarily massive, but it certainly is increasing, and it's an increasing problem for our beekeepers, these losses. Why? What's going on? We have a fairly good survey of beekeepers in terms of, well, what problems do they recommend or suggest are problematic? And certainly Varroa is over half of all winter losses here in New Zealand, as it is, I suspect, in many places all around the world. We have queen problems, we have natural disasters in some years especially. We have suspected starvation, and yellow jacket wasps are really a problem in some years, especially warmer, drier years. It's a considerable problem here. These problems, as I said, we have increasing rates of colony loss, winter colony loss, due to probably mostly Varroa. It's not quite what you guys are experiencing in North America at the moment. This is data from quite recently. huge losses, average loss of 62% in this particular survey in the US, and I understand Canada's, large portions of Canada is being affected by this as well. And certainly it seems to be a crisis level, and it's likely that Varroa is a considerable problem and cause of that losses in your country as well. Pesticides, right? So we're getting into synthetic chemical pesticides. These are really key in New Zealand, as they are everywhere around the world at the moment for the control of Varroa. Lots of us are working towards non-chemical control, the use of non-synthetic chemicals for Varroa control. And it would be lovely to imagine that we're going to have a resistant bee strain here in New Zealand, and we're going to have all sorts of other approaches that are going to be really successful in the control of mites. But I suspect over the next little while, it's true, unfortunately, that we are going to have to use synthetic pesticides for a long time yet. They're an essential component of our mite management regime, and we are going to need to utilize them. Certainly Amitraz, Coumaphos, fluvalinate, formic acid, those are the key ones, you know, fluvalinate, Coumaphos, Amitraz. Pyrethroids are really essential for mite control. In New Zealand, I suspect we are similar to what many countries are around the world. So this is a sort of a Venn diagram of all the different uses and ways that mites are controlled. So flumethrin over here, Amitraz, and the big circle for flumethrin here indicating that a lot of beekeepers utilize it. Big circle here for Amitraz, a lot of beekeepers utilize that, a lot of utilizing oxalic acid. And I suspect this is similar wherever you sort of look at. So pesticides are a key component of our use, right? So there are other methods such as drone removal and formic acid and that sort of thing that's included in here, oxalic acid up the top, but these synthetic chemicals are really important. This is a survey of around about 9,000 beekeepers. So a pretty good survey. We've got a pretty good handle here on what beekeepers are doing to control mite populations. And what is worrying to me, most worrying to me, is that there are some beekeepers, this 13% up here, that are using flumethrin only, right? So they are not using any other form of mite control, and 10% that are using Amitraz or Apivar only, right? So I suspect this sort of model is probably kind of typical everywhere. You've got some beekeepers in your community that are probably doing their own thing. There are probably some that are doing a really good job in terms of controlling mites through different ways. But it is the case that well, okay, 1,200 of those are beekeepers in New Zealand are using flumethrin only, and 900 are using Amitraz only, which is a problem in terms of resistance management. And that's what I'm going to talk about today. So integrated resistance management for acaricide use on Varroa destructor. It's a sort of a review that I wrote a little while ago, trying to get a handle on what's going on in the field and how we might improve our methods for resistance management. What is integrated resistance management? It's to prevent the evolution or emergence of resistance in Varroa. We want to keep populations susceptible. So we want to prevent resistance development in susceptible populations, slow down the development of resistance, or reverse it to a level that allows effective and efficient use of insecticides, acaricides in this particular model. And certainly we want to limit adverse environmental effects. I gave the key message from the talk away a little bit earlier, but it certainly is the case. I think that that Varroa is a really challenging pest for integrated resistance management, and we need to be careful of it, and we need to be much more careful of it than what we have been previously out there. So yeah, how do we do integrated resistance management? Well, there are three key methods. This is a French paper from 1994, which sort of summarized the field. And the three key methods involved sort of a multiple attack, right? So using multi-directional selection pressures, lots of different pesticides with different modes of action is the key there. Using rotations, using mixtures of pesticides or of unrelated insecticides that have different modes of action. So that's the really key one. And I'm sure you'll all have heard about the importance of rotations for your use in pesticides and Varroa populations previously. So that's the first one. So your multi-directional selection pressures, your multiple modes of attack. Second one is focuses on resistant individuals. So resistant mites in your population. How are we going to target better those resistant individuals? So can we have an elimination of the selective advantage of resistance individuals? How do we target them? Do we do things like have increased insecticide uptake through attractants? Maybe we could attract Varroa to the chemicals. Maybe we could suppress their enzymes that they use for resistance and detoxification enzymes. Maybe we could do something like that. And then the third approach to integrated resistance management is one where we acknowledge the value of susceptible individuals in the population, right? So how do we ensure that there are lots and lots of susceptible mites in our mite population? They're a valuable resource, right? So those susceptible individuals can interbreed typically and potentially dilute any resistance genes out there. We need to make sure we can keep those susceptible individuals out there and reduce the selection pressure for those populations. All right, so hopefully that's a sort of a general idea about what integrated resistance management is. It's the application of multiple selection pressures, different pesticides with different modes of actions or mixtures. It's the use of ensuring that we can try and eliminate selective advantage of resistance individuals, so to get rid of those resistant individuals, or manipulating the susceptible individuals and susceptible genes out there in the population. Okay, so that's what Varroa is, a super pest, right? It's everywhere. It's a really big problem. It's got resistance to lots of pesticides, and that's what integrated resistance management is. What I wanted to talk about next is before I really get onto the methods of this, is two key problems with integrated resistance management in regard to Varroa populations. And there are two major problems. And this is the first, a lack of fitness effects from what we know so far. A little bit of basics, right? Going back to how pesticide resistance develops in populations. So typically in populations, what you'll do is you'll have a resistant population, resistant individuals in the populations, right? Initially, your individuals, the pest on a leaf might be, dominated by these susceptible ones that are here. You might have only 1 in 100 million resistant individuals in here. You put on your pesticide application, you spray it to your leaf, you kill off lots of the susceptible ones, obviously. A few susceptible ones are left, and then the resistant ones are in the population. And they breed, you apply that pesticide again and again, and it's acting as a selection pressure, killing off the susceptible ones, and the resistant ones are developing until you end up in scenarios where you have lots and lots of resistant individuals and very few susceptible ones, right? This we've seen this again and again and again, whether you're looking at weeds, whether you're looking at mites, whether you're looking at whatever, right? So there's genetic variation out there, and there's random mutation that causes these resistant pests populations, and it's selected for effectively over time. So pesticides act as this massive selection pressure in these populations, especially if we use the same chemical over and over again. The one key sort of dent in the armor almost of the resistant individuals typically is that there's a fitness cost associated with being resistant. So those resistant individuals, such as this one here, may have sort of lower fitness, less reproduction, that they might not be able to perform quite so well in the population. So we see that again and again. So if you take the pesticide away, then the other ones resurge effectively, that they become more abundant. What is it with Varroa? Well, if in normal populations, if you see resistance and take away a pesticide, then that Varroa populations might revert to susceptibility over time. And for that susceptible population reversion, going from resistant to susceptible populations, if you take away a pesticide, then you need some sort of fitness effect that's going on in these populations. And do we see that? There's actually very little data out there that suggests that actually occurs with Varroa. There is one particular study, Stephen Martin, who's a legend in this area, done a huge amount of really good work. Right, his work in a Texas population early on found that asked the question, well, do pesticide-resistant Varroa have lower fitness? And found actually it appears that there is little or no reproductive fitness cost associated with pyrethroid resistance in Varroa, at least in Texas. That's a depressing piece of news, unfortunately, for Varroa management. Right, so we can't just take away a pesticide and imagine that fitness is going to revert. Right, so this is a big problem. There's actually very little other data, unfortunately, out there that has been worked on for other pesticides. So we don't know for amitraz or other pesticides if the same thing occurs, but certainly it's a problem here for Varroa populations. The other issue, incestuous reproduction, is what I've labeled it here, right? So lots of you will be familiar with Varroa reproduction and how they go on. This is a bee pupae here. I pulled it out. I put it on the side, and it's an early stage pupae. Here's a Varroa foundress mite. Here's her daughter, and here's the male over the side here. So lots of you will be aware of what goes on in populations. Ideally, for Varroa or any pest, we'd like to have, okay, we can see resistance decline in populations if you have susceptible individuals that mate with resistance. But this doesn't really occur a lot necessarily in Varroa populations. They have an incestuous mating system. So unless you have really high populations of mites. So many people will probably be aware of this, right? So when you've got a queen, she lays an egg in a hive, right? So that egg hatches, turns into a larvae. That larvae and its later instars will become attractive to Varroa mites, which will crawl off that bee and go into the cell and hide down in the brood food down a little bit there. She's alone. So she's in there by herself, right? If there's really high populations, there might be 2 mites, but typically, if you've got really good control of your Varroa, then you've just got 1 Varroa mite hanging out in here. What she will do is she'll lay some eggs, right? First egg is a male, second eggs are fertilized, they are females, the brother mates with sisters. So this is this incestuous reproduction that's going on here. So effectively what you can do is, even if you've got really good mite management, but a few mites, then you've got this incestuous mating system that means that there's no dilution, there's no mating of susceptible and resistant populations going on in here, right? So you're just maintaining whatever genes that are in there are typically maintained in that sort of population. So you might be a good beekeeper. You might be a wonderful beekeeper who manages your Varroa down to really low levels and keeps them at low populations. But if you are doing that, then you are limiting the potential for susceptible mites to mate with resistant mites, right? So you're not allowing multiple mites per cell. You're not seeing interlineage mating, and you can potentially be maintaining your resistance in these populations. So those are the 2 key problems with Varroa that I see. in terms of integrated resistance management. We've got this scenario where you've got incestuous mating, and you've got what we know of so far is a lack of fitness effects. We're not sure it's occurring with all pesticides, but it does seem that susceptible mites are reproducing just the same as resistant mites in these populations. What I'll do next is really get on to, well, okay, given these issues, given that the mites are such a problem, what can we do? What can we do about this? How can we use integrated resistance management? And I'll go through each of these scenarios that I've talked about before. And this first one in terms of involves the application of multi-directional selection pressures, right? So rotations are especially important for an awful lot of pests out there. Are rotations actually useful for Varroa control? And can we utilize a range of different insecticides that have different modes of action for Varroa management out there? So can we do this? Can we utilize pesticide resistance to revert resistance? So I'm specifically talking about reversion of resistance here. It is certainly the case that our mite populations around the world have developed resistance to pyrethroids, to amitraz, to all our coumaphos, to all our key ones, our pesticides that we're utilizing. So for strategies such as a rotation to revert resistance, right? So this is changing your amitraz resistant mites back to susceptible populations. You need different pesticides with different modes of action. And yeah, we have that, you're right, in North America, and we have pyrethroids, we have amitraz, we have coumaphos, we have different modes of action going on there. But for this reversion of resistant to susceptible populations, you need to have a fitness cost typically associated with being resistant. And what I've just talked about, okay, there's a lack of published evidence that there is any resistant cost to Varroa, right? So it appears that there's little or no resistance cost to being resistant for Varroa populations. So what does that mean? Well, out there, it means that it's likely that reversion from resistant to susceptible populations is kind of rare. We shouldn't really be seeing that necessarily a lot in populations. We do see it apparently occasionally. So this is some work from 20 years ago, over 20 years ago, where in Italy, where they were looking at the decline of pyrethroid resistance over time. Pyrethroid resistance probably evolved in this neck of the woods in Italy and places. Right, so the decline of pyrethroids was likely, they concluded, to take many generations. They were suggesting that rotation would only be useful for pyrethroid if it was used only once every 4 to 6 years, and then immediately by another chemical. And actually, in terms of reversion to resistance, there's much more common work out there suggests that rotation is not necessarily for reversion of resistance. This is an article from South America looking at coumaphos resistance, and considering that coumaphos had not been used for 9 years in the particular apiary where this study was undertaken. The results provided here lead to reflections about the problem of resistance phenomenon and its population dynamics. They didn't see after 9 years of not using coumaphos, it was still present in the population. So once we get resistance out there, then it's unlikely that we're going to lose it in a big hurry. So it's really something we need to avoid and control as much as we possibly can. How might we go about doing that? How might we best use insecticide or integrated resistance management strategies out there? This is an article from the Journal of Theoretical Biology. A paper, it's a modeling study, but it's one of the better ones that I kind of like anyway, that asks the questions, well, how do we best manage resistance in populations? And they were concluding that determining the optimal strategy in resistance management was dependent on sexual or asexual reproduction. And they unfortunately concluded a rotation of 2 insecticides in alternate years was only really the optimal strategy. So we've got to work on this. And actually, mite biologist Rosencrantz et al. In 2010, so these people know a lot more about mites than I ever will, suggesting that the mainly non-professional structure of beekeepers community has implied that rotation may have only ever been a short-term solution, at least in resistance management. So we've still got to keep on doing it, I think. We've still got to keep using rotation. It typically, for lots of pests, slows resistance development for many pests. We've really got to limit the resistance development. Reversion will be hard, is the conclusion from that. So totally, those 13% or 10% of beekeepers that are only using flumethrin in New Zealand or only using amitraz, we've got to encourage them to utilize resistance, right? So typically we've got to use resistance rotation and slows resistance. Please utilize it. What else can we do, right? So this paper, I'll keep coming back to it again and again. So this paper was one that suggested, well, okay, a pesticide mixture would be best if an insect reproduces sexually, and sequential application might be best if asexual, but reduced dose mixtures and rotations being optimal in some cases. So I think there is evidence out there to suggest that mixtures might be useful, reduced dose mixtures might be useful. But at the moment, this is an area that we need more research in before we can utilize it. So I wouldn't recommend mixtures for any management at the moment. Certainly rotations are optimal in some cases and maybe good for stopping the development of resistance or limiting the development of resistance in mite populations and do that. So certainly pesticide mixtures have potential, right, and there. And there are potential for some of that, some research out here to suggest that well, okay, utilizing one pesticide can have a diversity of effects, can offer a pull-push scenario. If you have a mixture of pesticides, it may be effective in terms of managing resistance, but it certainly needs more research before we can get into that and understand that a little bit better. The next thing that I wanted to talk about was management by saturation, right? So this is really a focus on resistant individuals out there. How can we target resistance individuals, right? And an awful lot of that is in regard to, well, okay, can we attract resistant individuals to the pesticide? Can we suppress their detoxification enzymes? Can we limit those genes that are effectively giving them resistance out there? And there's an awful lot of this work and in the next one, which needs more research before we can do anything. But certainly there are suggestions out there in the big world of pest management that there are approaches that could be utilized. Gene silencing being one of those. So RNAi, this is not a genetic modification in terms of altering the genome or anything, but gene silencing can potentially stop the expression of detoxification enzymes and is a useful approach for that double-stranded RNA technology. Perhaps we can utilize double-stranded RNA technology to target resistant genes and increase the susceptibility to other insecticides. That's possible to stop the expression of detoxification enzymes and proteins out there. I think that's a really worthwhile area of work. There's lots of pests for which we have pheromone attractants, right? So there are lots of pests out there that we can attract the males or females to a particular area and kill them. We've got no attractants for Varroa, and we've got no synergists, chemicals that can increase the efficacy of our pesticides yet either, unfortunately. So it's an area that people are working on. It certainly is a scenario in lots of other pest populations that can be quite effective out there for brown rice hopper, plant hopper on rice, but not necessarily in Varroa yet. Management by moderation acknowledges the value of pesticide susceptible genes and populations and reducing the selection pressure. All right. So how do we lower the selection pressure? How do we increase the number of susceptible genes out there? And a common way that we utilize in pest management to be able to do this is to have a non-chemical control methods. Right there, and certainly with Varroa, that is something we've got out there in a big way. So this is a paper that I kind of like, 3 pillars of Varroa control, right? So you've got the epitechnical measures, you've got the chemical treatments, you've got resistant stock. Resistant stock includes honeybees that are bred for resistant to Varroa. There's lots of success for that around the country. I talked to some beekeepers who tell me that in North America and places that you don't need to select for have some chemical control in their populations. They've got resistance now, bee strains that are resistant to Varroa. Epitechnical measures include drone brood removal, right, and trap and comb techniques and approaches like that. But certainly chemicals are a big part of that. So utilizing epitechnical measures along with resistant stock is great beekeeping practices that hopefully contribute to our sustainable approaches. We do need in the sort of other approaches and other ways to control alternative hard or soft pesticides, including biopesticides. Oxalic acid is an increasingly used treatment here in New Zealand. Lots of beekeepers are utilizing that with a lot of success, and some are only utilizing that as their success, as their mite treatment here in New Zealand, on varying levels of success. There are other things that are coming on the market, and including biopesticides by using gene silencing. Here's some work that we've been doing with Greenlight Biosciences in North America and in the US. Suggested it might be the perfect pesticide, and our work is here in New Zealand has suggested that, well, it's really an approach to suppress Varroa reproduction in honeybee hives. Here's some bees that are effectively feeding on it. This is a product that's ongoing and under registration in the registration process in the US, and there are trials that Matthew can talk about a little bit, and I can talk about it a little bit more later here in New Zealand. There are trials in Canada as well. So it's a product that might be available. It's a biopesticide, and it's potentially a soft pesticide, a highly targeted, hopefully, pesticide that we might be able to use. And we can talk about that a little bit later if you want. What I'd conclude, coming to the end of what I have to say, is really that we must and can do, we must do better to achieve integrated resistance management with Varroa. The industries as a whole must get together and really get around to doing this. We've got to really ensure that our synthetic pesticides are used in a way that promotes their efficacy and longevity. There's things like the double-stranded RNA. There's all sorts of resistant bee strains out there that are going to be useful out there, and we hope that they are going to arrive on the market really soon and going to solve our Varroa problems. But in the next foreseeable future, synthetic chemicals, synthetic pesticides are really important, and we've got to do better as an industry, as users of these, to promote their efficacy and longevity in these populations, and we're not doing that. We can look at what other groups and other pest managers are doing around the world, and this is one that's not for Varroa. This is for mosquitoes. And this is looking at malaria management, right? And insecticide resistance management, integrated resistance management in malaria populations. What they are doing is having short-term, medium-term, and long-term goals, right? So they're trying to preserve the susceptibility and spread of resistance, all right? In mosquito populations, they're trying to do things like improve our understanding of insecticide resistance for it, and adapt strategies for control, and long-term trying to use innovative solutions, right? So I think as an industry, I'd really encourage us to globally almost get together, and certainly in a big place like Canada, get together and figure out ways of doing this. how do we manage our insecticides a bit better? It does have implications in management of this, and this is some work that I found actually this week, right? So a large proportion of beekeepers around the world don't adhere to treatment regimes with major consequence, right? So this is a study out of the UK where they're looking at adherence to treatment policies over there, right? And what they are finding is that the lower overwintering colony losses are predicted by correct treatment timing and effective use of chemicals. And they're suggesting that treatment non-adherence, right, utilizing the wrong products or illegal products or doing it at non-recommended times is a really large driver of colony mortality over there. So as an industry, I think that the key lesson from this sort of study is we need to get together to work out some best management techniques and how we do that, and implement them across the sector and hopefully limit our resistance. This paper actually is a really interesting one. Some data out of it, right? So they're looking at things like winter colony losses, ranging from really low to really high colony losses, and beekeepers that were utilizing a whole bunch of different approaches, and things like flumethrin and amitraz are really essential for mite control and associated with low colony losses. Whereas, you know, using icing sugar somehow to control your mites is not effective in higher colony losses. And that also comes through in terms of honey production, right? So honey production in terms of low to really high honey production. The highest honey production is associated with the use of synthetic chemicals such as amitraz, but also, you know, drone brood can be one of those three pillars of Varroa management can be utilized as well. It's quite effective on there. So if you want to increase your honey production, have good mite management, and clearly synthetic chemicals are critical to that. My conclusions, right? So conclusions for integrated resistance management for Varroa, right? So we need to avoid resistance if we can. Reversion seems to be with Varroa populations because of their incestuous breeding and because of lack of fitness effects that we know of so far of being resistance. Reversion is likely to be really hard out there, right? So it's going to be quite difficult. Data and monitoring is key. So science is key. I encourage supporting your science, supporting monitoring for resistance, supporting mite abundance monitoring out there. So understanding if and where we have resistance developing is sufficient to interfere with effective mite management is really important. We've got to get a handle on that, right? So understanding where resistance is developing. Rotation is probably a good thing, at least for trying to avoid the development of resistance, and a huge supporter of rotation. We've got to rotate your chemicals. It seems like this is a really important thing to do. Keep doing it, but certainly reverting to from resistant to susceptible populations, rotation may not be all that useful. So try and stop resistance development if possible. We need alternative products out there, so we need the scientific community to get their butt into gear and do a bit more, perhaps, in terms of utilizing biopesticides like double-stranded RNA or oxalic acid or resistant bee strain populations, right? And I think there's research out there is really needed in terms of approaches such as utilizing mixtures or attractants or things like that would be really ideal for us in terms of trying to figure out better and more effective and having more tools in the toolbox for Varroa management. All right, I think I've probably said enough. There's a nice picture of us in New Zealand doing some beekeeping. So thank you again for the invitation to speak, and I hope that was useful.
Thank you very much, Phil, for your very interesting talk. And I think we have already received quite a few questions here. We actually received some of the questions in the Q&As and some questions in the chat. So what I'm going to try to do now is order them by time. So I'm going to read the first questions first, as much as I can. So Paul Gregory had a question. Do you recommend early autumn feeding to reduce brood rearing, allowing miticides to work in a more efficacious manner? In Canada, we can plug out the brood chamber to shut down brood rearing.
And people definitely do that, right? So here in a brood break, effectively, and whatever way you can do a brood break, it can and has been utilized as an effective approach. It's quite labor intensive to be able to utilize in a lot of scenarios to be able to do that. If you're a beekeeper with several hundred hives, that can be quite an effort. But can and it has been having a brood break of some sort has been an effective way to do that. I think that was the question.
The next question is by Cheryl Harrison. She would like to know, is resistance more prevalent with man-made chemicals, or does it matter? For example, will Varroa develop resistance to oxalic acid, a naturally occurring substance?
Yeah, so we are yet to see any evidence anywhere really that mites have developed a resistance to oxalic acid. A slight issue with oxalic acid is we're not quite sure of how it works. There's lots of theories out there and that sort of thing about it burning the mouth parts and all sorts of things on how oxalic acid works. And some of the other experts online might be able to correct me or have their own theories. But we don't necessarily know how oxalic acid works for mite control. So monitoring for resistance and understanding how resistance might develop is hard. But at least so far, to my knowledge, there's no evidence anywhere of any resistance to oxalic acid. I'm not expecting it in the same way that we would for synthetic chemicals like amitraz or pyrethroids.
Because you think it's more difficult to develop resistance for the mites against these more physical mechanisms, like a physical burn or...
Yeah, a little of both, right? So I think there is certainly that aspect to it in terms of it may be quite difficult for it to gain a thick exoskeleton or something, something like that. But also, if we look at pyrethroid resistance in places like Italy, pyrethroid resistance was found really soon after mites became a problem over there, and oxalic acid has been utilized in a lot of countries so far for quite a while, and we're yet to see any evidence of any resistance in those populations. So I think it's a combination of if it is a physical scenario where the mouth parts or elements of the mites are being burnt by the oxalic acid, then it's going to be harder to develop resistance. It's still possible. You could still imagine scenarios where there might be mites that develop an avoidance mechanism, behavioral resistance, effectively. So anything is possible out there, and mites are a great example of a pest that's done some interesting things to avoid us controlling it. I'm hopeful that we are not going to see oxalic acid resistance out there.
Next question arrived in the chat. It's by Ian Stepler. Hi, Ian. His question is, can you comment on the possible harm rotating chemical treatment has on colony health, synergies from mixing chemical residues?
Yeah, so that's certainly an area that needs a bit more work, right? So the rotation of chemicals, right? So we know that there can be some negative effects on chemicals of chemical use on your bee populations, whether that's or on any pest if you're utilizing chemicals, whether that's on their microbiome or whatever. Right, so some of the pesticides we use can be hard on our bees out there. And the residues can be seen in populations at relatively low levels, at high levels in some situations. So I forget what the question was, but I think that there are issues with residues, chemical residues in populations, they may have an effect. I think Varroa has a massive effect if we don't control it. So synthetic chemicals are going to be needed, unfortunately, even though they might have some effect on Varroa, they might be needed in mite management for some time yet. Ideally, we'd stop using synthetic chemicals, we'd stop putting them in our beehives, we wouldn't have to worry about that, but I think in the short term, we need synthetic chemicals for mite control.
Next question from Riolito. Hi, Delan. I hope I pronounced this correctly. I'm not an expert. I just learned that Varroa originated in Asia. What are your thoughts on Varroa activities, differences in tropical and non-tropical countries? So
I think the non-tropical and tropical countries, that one of the biggest effects that we see here across New Zealand to an extent is that in warmer parts of the country and in warmer parts of the world, you don't get a brood break. So that's probably the biggest problem that we have in different countries, different parts, different regions of the world with different temperatures in terms of, well, Varroa populations here in New Zealand, especially further north than me, they've got brood to breed on all year round, and that's a big problem. If you have a brood break, then you, like you do in Canada, then you might be able to utilize different approaches to managing your mites a little better. For the most part, though, you're thinking about a Varroa in a beehive. Bees are wonderful creatures that maintain their colony at a pretty set temperature, right? So they're trying to keep it in a pretty constant temperature. So for them, it doesn't really matter a whole lot, right? So the mites are exposed to a beautiful warm environment year round, effectively in many parts of the world. So yeah, it doesn't matter to a mite if your beehive's in tropical area or a subtropical, except with the brood break area.
The next question is actually a question that I had in mind as well. Marta Guana is asking, you mentioned the lack of reproductive disadvantage from mites resistant to pyrethroids. Based on the different mechanisms of action of pyrethroids and amitraz and the different timeline for resistance development of both compound types, is it possible that there is actually a reproductive disadvantage on some of the amitraz or on amitraz resistance in Varroa populations?
Yeah, and the answer is yes, absolutely. You know, there is that possibility that there is a fitness effect that's going on in those populations. So somebody needs to do the work, right, to really understand it, so we can do resistance management better. But that study by Stephen Martin's group was a rare one. We need to understand more about how amitraz affects the fitness of these mites. My suspicion is that there's not a fitness effect given the maintenance of the resistance in populations, and especially in the US. But that's a wild guess that's not based on any scientific data, which is bad for a scientist to be promoting. So the answer is yes, your person is absolutely correct. It's entirely possible that there is a fitness effect for amitraz resistance. We don't know. We need to do that work to find out.
So I will allow a few more questions. So the next one from Matthew Polinsky, and I think it kind of is related to one of the earlier questions. We know that beekeepers tend to try various applications and mixes. Can Phil comment on research on pesticide mixtures for Varroa, mixing various treatments simultaneously? What is Phil's view on the research being done or that needs to be done on this? Furthermore, what are the challenges in getting this done?
Sure. So my wife works for the Ministry for Primary Industries here, and is involved in pesticide management, and she would hurt me should I promote the use of mixtures that are off-label, right? So what I would suggest that everybody does in Canada is utilize the pesticides as they are on label. So utilizing mixtures and those approach, I think has a lot of potential and a lot of hope. We need to do a lot of research on that use out there, and before we do that, I would not promote the use of mixtures, or I'd get in big trouble. So please don't do that. I think it's got a technique that's got real potential. I've heard unofficially of some beekeepers utilizing such an approach and having success. But beekeepers tell me all sorts of things at times, so I'm not sure how accurate that is. We need more research before we can do that.
The next question is from the chat, from Anthony Antonucci, and he is joining from the US, if I see this correctly. So, but I'm not sure. He says there are similarities in pests between the different countries. He is wondering about Tropilelaps, and researchers from different countries and different regions co-joining in their efforts to research Tropilelaps mites in order to be more proactive before Tropilelaps really hits in North America. So I guess he just would like your comment on this, if there is some sort of collaboration that you're aware of.
Yeah, so people are starting to panic a lot about Tropilelaps, and I think with justification. So I understand that it's managed to cross its way into at least the Ukraine from Asia, so it's got that far. And once it's in Ukraine, it'll spread probably across Europe, and people are doing things like importing beehives and that sort of thing, bee colonies. It's going to be a problem. So I think we as an industry need to think about this, try and limit its spread and movement. I think it's going to be a massive problem. There's a number of researchers out there who are suggesting it could be worse than Varroa. So it's certainly something to keep on the horizon as a potential pest. We aren't here in New Zealand, to my knowledge, involved in any work, but there are a number of North American groups, especially out of the US, who are actively working in Asia to try and understand better control methods and more about the mite biology. So I think it's a real problem, something we need to keep on the horizon and be aware of and be thinking about.
Last question, is there any resistance on the horizon for formic acid?
Again, not that I'm aware of. So neither of the acids, formic acid or oxalic acid, to my knowledge, that there is no evidence out there for resistance to those populations. I hear occasionally that oxalic acid has variable efficacy in populations, but I think that's commonly around the approach to its application rather than any resistance in those populations. So people apply formic acid or oxalic acid in different ways and with different efficacy. I think it's just the application method more than any resistance in those populations.
Thanks a lot for all the questions. Thank you, Phil, for answering some of them. I don't know, maybe we will find a way to answer the remaining questions with a little bit of delay. Right now, we are going into a 5-minute break. But for those of you who don't want to take a break, you can also still follow the conversation here between the members of the tech transfer team, if you would like. Otherwise, see you in 5 minutes for the next presentation.
Thank you.
Hi everyone, my name is Medhat Nasr, and I'm the tech transfer lead for Saskatchewan beekeepers. in this roundtable discussion about Varroa mites. And today we have two other team members, and one of them is Andrew Byers. And Andrew, can you introduce yourself?
Thank you, Medhat. Yeah, I'm Andrew Byers. I'm the tech transfer lead here in the Atlantic region, and just pleased to be part of this great event. So we just wanted to spend a few minutes during the breaks to give people an opportunity to ask some questions or raise some points, or for us ourselves to talk about issues related to Varroa or important things in our industry in our province or region.
Okay, thanks, Andrew. And Matthew?
Hi, everyone. Yeah, I'm Matthew Polinsky from Manitoba, and I'm the program lead for our tech transfer here. Thank you to Phil for the excellent presentation. At the end of the kind of day today, we'll have more time to go into general discussion, but while we're having a couple minute breaks between presentations, we can kind of chit chat a little bit, or perhaps field a couple questions.
Actually, in Canada, we have 7 programs across the country. Each province has one, except Maritime, the 3 or 4 provinces you consider, that's one program. So our mission actually to basically we work through the industry, and we do the applied research to address problems of today in the field, and to try to develop recommendations based on our regional differences, or based on the bee management practices in each province. So that really helps to provide what we call a designed recommendation to fit each province in terms of bee health. And most of our research actually is being focused in the last few years on Varroa mites. So having said that, I think we are quite lucky to have this program started in 1994, and it's been around almost for the last, since that time. And each province work hard to make sure each program is being funded through the industry itself or government grants to continue serving the industry and address everyday problem. And if anybody wants to add something here.
Yeah, I think.
Sorry, you go ahead, Matthew. Sorry.
I just wanted to add that it's as Medhat mentioned, we respond, and our work is in line with the needs in our different regions, and having this national conversation is really important to hear about everyone's challenges and learn together. But also we need to always keep in mind that we are coming from different regions in a lot of cases, so we're going to be managing things slightly different seasonal changes. So something to think about as we hear all the different presentations today, how we can adapt it to our locations. Go ahead, Andrew.
Oh, that's pretty much what I was going to say, Matthew. The strength of our collaboration with the 7 tech transfer teams across the country is that we think similarly, as obvious by our comments being aligned. But what we can do is we work together quite closely. I spend quite a bit of time with Medhat and Matthew discussing the problems of the beekeeping industry across all of Canada. But the solutions sometimes require a national approach, but oftentimes they don't. And that's where we can work collaboratively to talk about these issues, come up with answers, but then we have to adapt that and make sure that it fits for our province and our region. Because listening to Phil's talk, and as I say, in our frequent discussions with the other tech transfer leads, I realized that there's great ideas out there and some very intriguing things mentioned in the previous presentation, which we'll have to think carefully about. But then as the provincial or regional tech transfer teams, we apply that for our own beekeepers, which is a really important aspect of it. So a real strength of this network of tech transfer teams across the country to be able to look at things broadly and then bring it home.
There's a question for you here. Are any of the maritime provinces free, Varroa free?
Well, I never miss an opportunity to encourage everyone to help keep Newfoundland Varroa free. So at the moment, and I'm going to go out on a limb here, I think that we have one special case, a little pocket in our own country of the island of Newfoundland that is Varroa free. And we really do all we can to make sure that it will stay that way, because not that they're not concerned about Varroa in Newfoundland, because they are, and they want to be prepared for it if need be. But in the meantime, we want to make, whatever efforts it takes across the country and internationally now to keep this one small isolated area of Varroa free. I think it's important, certainly hugely important for our Newfoundland beekeeping industry, but also important globally to have that little pocket of Varroa free beekeepers.
Thank you so much, and I'll turn the discussion to Ulrike.
Yes, thanks a lot. I hope everybody is back from our short break or has listened to the discussion with interest. And now I would like to introduce our next speaker, Geoff Wilson. Geoff is Saskatchewan's provincial apiculturist. He is supporting beekeepers in the field with mite management, best practices, and biosecurity. He also keeps his own bees to test new management strategies. And today he is not joining us from Saskatchewan, but from Thailand. So I am really grateful that he stayed up this long to give his talk live. Thanks a lot, Geoff.
Well, thank you very much for inviting me to speak. It's much appreciated. I have had quite an adventure getting to this talk today. Just before leaving for Thailand, all my baggage was stolen, carry-ons, everything, including my laptop. So I've lost all access to my presentation that I had previously made and had to come up with a brand new one within very short order. So the presentation's not going to be exactly like I had originally intended, but it's going to give very, very nice points. We are going to first talk about what's happening in Saskatchewan, how the teams work together, or the various groups work together serving beekeepers. And then we're going to talk about a little bit on management plans and management practices. I should really thank Maggie Gill and her research team on Tropical A Labs here. They've been very supportive of me getting this presentation together, very helpful, loaning me equipment and getting things in order for me to be able to speak here tonight. So a little bit about Saskatchewan's background for those that are out of the province. We have about 1,350 beekeepers with approximately 100,000 colonies. In addition to the 100,000 colonies, we have about 35,000 nukes. So in the winter, we put into winter about 135, we'll call them bee units, 135,000 bee units. Running those colonies, we have pretty much 120 commercial beekeepers that operate 90% of the colonies and 95% of the nukes, because our small scale beekeepers aren't making the nukes the same way that our commercial beekeepers are. We have very similar climate and production and overall beekeeping situation with Alberta and Manitoba. So the prairie provinces are very similar on our beekeeping experience. So Varroa, we know, is the biggest biological threat to our beekeepers. So it's the biggest problem we have that we can manage. We've got other problems like honey prices, what's happening with international trade, labor. But for disease management, Varroa is the key issue. When we're designing our management plans, we have to think about how we're going to approach the problem. Really at the center, at the heart of this is the beekeeper. It's the beekeeper that is managing the bees. It's the beekeeper that's going to be doing the work to make the bees healthy. It's the beekeeper that's going to find the problem and make sure that they understand what's going on and they do what the bees need. And then the rest of us are all support. So the tech transfer program, the researchers, the government, we're here to support. We all want you, the beekeepers, to succeed. So the focus has to be supporting those beekeepers in maintaining healthy colonies. However, at the same time, the communication has to be equal. We need to have trust among all parties, and we need to have feedback. So when we're developing Varroa management plans, the beekeeper is definitely the center, but the beekeeper needs to be in contact with the tech transfer programs, researchers, and government to tell us what you need, to tell us how things work. And then the government has to be in touch with the beekeepers and the researchers and tech transfer programs to help with that extension, to help put the programs in place, to help just get everything in line for the beekeepers to do the best job they possibly can. Tech transfer programs are wonderful. They get to do a lot of extra work, getting hands-on with the beekeepers, doing applied research, doing the things the beekeepers need. And once again, you got feedback from the researchers, being, in our case, the Western College of Vet Medicine, and the government and the beekeepers, in order to support what the beekeeper is doing. Researchers, same thing. They're a little further down further up the chain in research, where they are doing more primary work. But once again, a lot of it has to come from the needs of the beekeepers to best serve their needs. We have to have a lot of trust in between these groups. One thing in Saskatchewan, and I'm sure it's true in other places, we have been very successful in building that trust and keeping beekeepers, with tech transfer programs, government, and the researchers working together very collaboratively, telling everybody what they need to help bee health. The other aspect, it's very important to be proactive. It's a heck of a lot easier to be reactive instead of proactive, but it is much better to be proactive. Basically, what we're saying is be ready for the change before the change happens. So we can see that resistance is developing and we need new products. So where are we going? What are we going to do in order to have these products before everything fails, as a prime example? We have had all parties in Saskatchewan being very proactive. They've been excellent at this. Beekeepers, Saskatchewan beekeepers have been proactive for ages. They've started making nucleus colonies and wintering nucleus colonies for years. Our nucleus colony wintering is as good or better than our wintering for full-size colonies. Last year, we had 18.1% winter mortality for our full-size colonies. We had 18% winter mortality for our nucleus colonies. So that is a great resource. The beekeepers are making the next year's new colonies and are able to use them and have them in hand. In our case with the government, we had tested for resistance for apivar and Varroa mite levels since 2010. So we didn't start in 2008 when it was first registered, or 2009. 2009 was the first year I got here, but the second year I got here, 2010, we started testing for resistance for apivar. Then we stopped as soon as we found it at sufficient levels. So about 2021, we stopped testing for apivar resistance. And we realized we had an issue. And instead of repeatedly proving that we had the issue, we decided to address the issue and help beekeepers adapt. So we were advising beekeepers, instead of testing your mites for resistance, find another way to treat them. Tech transfer program and the research group, they're always working on what's next. So their whole goal, their whole reason for being is to be proactive. And the next step is repeated and consistent messaging. So we have basically the same message from the tech program, research groups, and government. We all promote the same exact ideas to keep your bees healthy. If a beekeeper calls any one of those three, they're basically going to get the same advice. And then that advice has to be repeated. But with updated material and information. And a lot of that information is coming from feedback from science, registration, and definitely the beekeepers. The beekeepers are key in us understanding what's working, what's not working, and how we do things better. Good example of repeated, consistent messaging. The beekeepers in the province may not realize it, but for the past 5 years, I have given the exact same talk about mite control. All 5 talks were completely different, but they had the exact same messaging on how to control mites in the current situation. So changing with the times, changing with the resistance development of apivar. So it's always been updated as we go. So different approach, same message. How well is it working in Saskatchewan? Well, we've found that we're doing quite well. The best we can do is compare with our neighbors, and we're finding that we are matching or exceeding what the neighbors are doing. You'll see in this chart, the province with the best winter is marked with green, the province with the middle wintering results is marked with yellow, and the province with the worst result is marked in red. All of this data, you can look it up yourself, is available through the CAPA winter mortality surveys that are published online. So it's just pulled from there. So you'll see in the past 10 years, Saskatchewan has always been first or second. And in the last 5 years, 6 years, Saskatchewan has been the best wintering, sometimes very significantly. For example, last year, when the neighboring provinces had 33.7 and almost 40% winter mortality. Now, saying that, we are seeing worrying trends. Every province, including Saskatchewan, has a slowly increasing mortality. So over the years, our average mortality is increasing. Last year was a little exception for us when we had exceptional wintering, but it keeps on increasing, which is a huge concern. So this suggests that the Saskatchewan beekeepers are doing a good job in managing their mites, and hopefully it's a result from everybody working together collaboratively and getting the best outcome for bee health. So we're going to go into a little bit of managing multi-resistant mites, and we're going to really focus on how some of these applied products work. So previously, our Varroa control was very simple. In the spring, beekeepers used apivar, checkmate, or apistan, placed them in the colonies, typically mid-March, and we got excellent results. Often that was the only treatment that was used. And then in the fall, we had a cleanup treatment. Occasionally, sometimes it was 2 oxalic acid sublimations. We use a lot more fumigation than we do drench. And sometimes it was a little bit of formic. And once in a while, it was some thiamol, but typically that spring single treatment of one of the synthetic acaricides was the key treatment. As you saw from the previous talk, this can cause issues with developing resistance. This treatment is quick, it's easy, but heavily reliant, most recently on apivar for mite control. So from last year, we noticed that mite control needs to change. We need to adapt. We can't do things the same way we have in the past. We are currently experiencing lower than desired mite efficacy for Apivar. Still working in many situations, still working very well in many situations, and it's working quite well in some, but other spots in the province, it's not working as well as it should. And so, because of that, it can still have a role in many mite control plans, but some of the weaknesses and some of the efficacy or loss of efficacy has to be taken into account. We also have organics. So we have the thymals, formics, and the hopguard, but these all have limitations. They're very variable. So according to the weather, according to the climate. The results this past winter has been mites were much higher than normal in most beekeeping operations. We saw much more visible signs of virus, particularly going into the winter in 2025. And we're anticipating for this spring coming up a lot more mortality in the province. I'm currently guessing that we're going to see our highest mortality ever in around the 30 to 35% mortality for the province. Hopefully, all the rest of your other provinces are doing better and you get much better mite control than what we're, sorry, not much better mite control, much better wintering mortality than what I'm expecting here. So when we are looking at our options, we do have the synthetics. And so all of the synthetics that we have ever registered in Canada are here. We have Apistan, which was the first product that we were able to use. We used it for approximately 8 years across the country, and then we ended up with resistance. Then we registered Checkmite, and Checkmite experiences varied. We had beekeepers reporting 4 years of active use, and then we had beekeepers reporting 1 usage before we had full resistance to Checkmate. So the resistance developed very quickly. We've moved into Apivar in 2008, and we're still using it now. It is not working as well as it has in the past. We are seeing signs that there are difficulties using this product with less efficacy, and that's in Saskatchewan, and it's also been reported in other provinces. So we do have concerns, but in many cases, it's still working quite well. And then we also have Baverol. Baverol was registered after Apivar. Unfortunately, when we tested the Baverol, we were never able to use it. The reason is it's a sister chemical to Apistan. The flumethrin and the fluvalinate work in the same manner. So when we got resistance to Apistan, and we developed the resistance to the flumethrin at the same time. We have done resistance testing after not using on bees and mites that haven't used Apistan since 2008, and that resistance has not gone away. So even though Apistan was not used for I guess it is 16 years now, we're still finding resistance to the synthetic pyrethroids. So what are organic options? We basically have essential oils. I'm going to claim that they are thymol. I do realize there's other active ingredients in Apivar, but the bulk of the active ingredient in Apivar is thymol. And then we also have Thymovar. We have organic acids, which are formic acid, being Formipro, mite wipes, which are multi-pad treatment, so repeated formic acid treatment. And then we also have oxalic acid. So the fumigation is the primary one we're using in the province, but we also have a little bit of drench going on. I realize the drench is more popular in other regions, for example, Quebec, but really our beekeepers have focused on the fumigation. I will highlight that oxalic glycerin is currently in registration process. However, it is not yet registered and not yet legal to use. And then the other option is HopGuard. I'm not going to talk a lot about HopGuard. We're not using it in the province very much. It's really difficult for our beekeepers to get. And so this is basically going to be the last time I'm talking about HopGuard, because we just simply don't have beekeepers adopting it. So I would like to remind everybody that you must use products according to the label. So whatever I say from now on, the labels change. If the labels change to change the way that it must be used, you must follow the label. So every time that you treat, make sure you're following the label. So right now, we have these organic treatments. They all have a lot of strengths. They also have weaknesses. We have to maximize those strengths. We have to treat the mites using these products, and we have to use these products at their greatest potential. And to do so, we need to look at the development of the bee. We have to look at how things work. This is a great chart. I hope a lot of you beekeepers have Beekeeping in Western Canada. It's a very nicely developed hands-on book. It is getting a little old, but it's got great information in it. So this was stolen or borrowed from Beekeeping in Western Canada. We have 3 castes. We have the workers, queens, and drones, and we have different development periods. We have the eggs, which last 3 days. We have the larva, which is the feeding period, and until it's capped, and it stays a bit of a larva, turns into a pre-pupa, and then a pupa. And then this is true for all 3 castes. The difference is how long each caste is in each stage. So we are looking at 21 days development for workers, 16 days for queens, 24 days for drones. Then we also need to look at Varroa reproduction. And this, once again, was adapted from Alexander. And basically, it's a really nice chart showing us the development of Varroa. We have the bee with the mite on it. We call this the phoretic stage. And so the mite feeds on the bee, and then until it decides that it wants to enter the cell. The mite moves underneath the larva, so this is pre-capping. It's entered the cell at about 5.5 days old, and then it hides right underneath the larva, hiding out in the feed. And after capping, crawls out from underneath and starts to feed on the pre-pupa. The female lays her first egg at 60 hours after the cell is capped, and then every 30 hours thereafter. The eggs start to develop from egg to larva to protonymph to deutonymph, and then to adult. It takes about 7 to 8 days for the female to mature and 5 to 6 days for the male. Mating begins within the cell, so they're mating brother to sister, which is really bad for resistance. It basically, if the trait gets into the population, it really amplifies that selection process because you have very inbred system. And then female leaves the cell with the mites, with the emerging bee, and once again runs around to once again infect new cells. Now, when we combine these 2 reproductive periods, we basically have mites entering pre-capping period, so about day 5 or 5.5 after becoming a larva, and then they crawl in and do their reproduction. So in the drones, they have 16 days that they're hiding out underneath the cappings. They're very hard for a lot of our products to hit. So we need to take that into account. For workers, we have a 14-day period. So because workers take less time to develop. For the rest of the talk, we're going to highlight the drones. Why? Because it is the longer development period where the mites are hiding out from the products the longest time. They're hiding out underneath those cappings. We also have a phoretic stage. This is kind of like a recovery stage for the mites. It's not absolutely necessary. The mites can climb back in and start laying eggs again, but they're not nearly as able to lay the eggs. They're not nearly as successful. So they need this period on the adult bees in order to be able to make more mites faster. This period is typically 5 to 11 days on the adults. So the bees want to be the mites want to be on the bees for 5 to 11 days. They typically pop off the nurse bees and they like to get on the day-old bees because the day-old bees are the day-old bees or newly emerged bees are second most attractive, and the nurse bees are most attractive. So the mites actually want to be on the nurse bees. So this makes a lot of sense. The nurse bees are hanging around the brood. When they're done hanging on around the brood, when the mites are done feeding, they have easy access to the larva and can easily get in and start their reproductive cycle again. The worker bees or the foraging bees are least attractive. There's actually a repellent called geraniol, which is a compound of the Nasr pheromone that stops the mites from wanting to be on the working forager bees. So they're very attracted to the nurse bees and day-old bees. And I want to highlight, when you are sampling for mites in your colonies, you cannot take samples for shaking from your brood, for your honey supers, from your lids. You need to take it from the brood chamber. You need to take it off a frame of brood. And that is to make sure you get the nurse bees and the day-old bees to get a very more accurate number. So let's add up that life cycle. Might spend 14 to 16 days in the brood, and they spend 5 to 11 days on the adult, called the phoretic stage. So when we look at where the mites are in percentages, we have at minimum 24% of the mites that are phoretic or easy for us to treat with a product. We have at most 31% of the mites that are phoretic. So basically, that's very simple math. 5 divided by 21. So 5 days at the phoretic divided by 21 days as a worker. At most, we have 31% of the mites are phoretic. 11 divided by 35. We have, that should be 25. We have mite growth. When we look at mite growth, this was wonderful work done in 2008 by Rob's Curry Lab in Manitoba. And we find that mites grow astronomically. They don't go addition, they do a multiplication. Mite levels start low and then they increase quickly. So if you start with a level of 3% in the spring, you will find your mite levels at about 80 to 90% in October. So they get out of control very quickly. I'm just going to do a little sidebar here from feedback from beekeepers who have said that the shaker jars aren't working, the testing's not working, for the sampling's not working the same as it has in the past. The tech team has developed a program or a research program to identify, basically redo this work under a new situation. So we think that the mites are probably more they're carrying probably more virus. They're having a bigger effect on the bees, and they may be needing new lower treatment thresholds. So we've got a program that our tech team is doing right now to determine what the new thresholds are going to be. So hopefully we get a new update on this chart that's more relevant for our current situation. It's more important than ever to sample correctly. Make sure you're sampling from the brood frames. I know I've already said this, but I like to repeat it because it is so important. Those nurse bees are key. You need to sample the mites where the mites are going to be. You have to be consistent. Always sample from the same place. Make sure you have enough bees. So don't take a tiny sample. Make sure it's about 300 bees per sample. This many bees will give a better estimate of what's going on. And then know what every mite means. So continuing, the treatment thresholds are continuing to drop. Remember that one mite in 100 bees is 1%, and know that what that means. Now, we always say if you're at 1%, plan to treat. So that used to be 3%, but anytime you're at 1%, plan to treat. In fact, now we've gone so far as to say if you're in 0.1% at the beginning of August, you're probably going to have to treat come September. So we need to understand what's happening. But yes, know what your mite levels are, and know that gives you the idea of how to move forward. So some of the products, we've got oxalic acid. This has become very, very popular treatment. This mite, when it's, this product, when it's fumigated, kills mites for about 3 days. When it's drenched, it kills for probably a little bit longer. 95% of our oxalic in the province is put on through the fumigation or vaporization or sublimation, whatever word you want to use. So I'm really going to focus on the fumigation here today. Remember, this only kills the phoretic mites. So we're killing only the phoretic mites for 3 days. So that means it's going to work best when there's no brood in the hive. One thing we're finding is oxalic can have a hard time penetrating very populous colonies or in the brood's nest. So we need to make sure that we're doing things in order to make sure the oxalic gets into the center. And some of that is temperature dependence. So you want to make sure you're not treating when it's too cold, because that cluster tightness affects how the oxalic acid circulates in the hive. We can get this product to work when there is brood in the hives, but we have to use repeated treatments. The best repeated treatment that we've come across, both through trials of my own and talking with a lot of beekeepers, is 5 treatments, 5 days apart, and pretty much 2 grams of oxalic per brood chamber. Beekeepers are reporting anywhere from 15% efficacy. Typically, that's when they only use the one oxalic acid treatment, all the way up to 95% efficacy with beekeepers using 6, 7, sometimes 8 oxalic repeats on that 5-day cycle. So how is this working? So we're going to look at our drone brood cycle, which is basically our 15 days. And our 15 days is on both of these arrows. And the top arrow is basically mites emerging, and the bottom arrow is mites entering new cells. So we have the mites that are emerging before the treatment, they're re-entering, reinfecting, and coming back out pretty much over at the end of this cycle. We need to break that. So we're with our repeated oxalic acid treatment. Basically, this first treatment is killing the mites. They're not re-entering. However, the oxalic acid runs out of power or runs out of its effectiveness by about day 5. So we put the second treatment on, stopping the re-entrance and killing the mites and stopping this re-entrance. Then Once again, the oxalic acid loses its ability to control the mites after about 5 days. So you put the third on on day 10. And you have to keep repeating. And this is the difference between drone brood cycle and the worker brood cycle. Basically, with the drone brood cycle, this fourth treatment, the mites stop coming before this fourth treatment. And with the drone brood cycle, the mites, there's another half of the treatment period that is not covered. So you need to have basically a fifth treatment minimum in order to use this kind of oxalic acid treatment regime. So tips and tricks for using oxalic with fumigation. Use at minimum 5 fumigations, 5 days apart. Do not do this with a drench. Drench no more than twice in one season. The drench is harder on the bees, it's harder on the queen. You will find more damage. We're really not seeing much impact on the bees when we're using the fumigation, which is very nice. And if you drench once or twice, you won't see a lot of impact on the bees. If you drench more, you're going to see smaller clusters in the spring. You're going to see impacts to the queens. Dosage, it is definitely registered at 2 grams per hive in Canada currently. There is a new registration going through, and it will be updated. And we do have trials going on in Saskatchewan by our tech team once again that suggest the new product should be applied at 1 gram for less than 8 frames, 2 grams for singles, and 4 grams for doubles. So basically 2 grams per brood chamber. I'm sure Medhat will be available during the discussion to talk about some of that work because he's the one that's doing it. The temperature is also critical to control. We find that a temperature of 190 to 200 degrees Celsius is optimal. Lower than that, and the system can plug up and takes too long to fumigate. And above the 200, we start to see the oxalic acid break down. So we like to keep it in that sweet spot to get good timing, cleaner applicators, and less breakdown of the oxalic acid. And then absolute key is do not sample for effectiveness until after the 5th treatment. So you've got these mites that continue to emerge, and the oxalic acid is working. You do need to get through the first cycle of the mites, or the first cycle of the brood, to clear the mites out of the brood before you sample. We're finding it much better to sample after the 6th treatment. We had beekeepers calling last fall. They sampled on treatment 2, treatment 4, and they said, hey, our mites are not dropping. They're not falling. Our numbers are not falling. In fact, some were saying our numbers are going up. I said, well, do 2 more and call me in the morning. And basically, they did the 2 more treatments 5 days apart, and just all of a sudden, the mite levels dropped. They came down to reasonable, and they ended up with nice mite control. So it's really important that you start to test to see if this kind of fumigation works after the 5th or 6th treatment. We also have Formicpro. It's another option. There's 2 options with this treatment. We have the 14-day treatment, which is 2 pads at once. And we have the 20-day treatment, which is 1 pad at a time, 10 days apart. Really nice thing about this product is it's currently registered for use with honey supers on the hive, which is a great option for midsummer treatments. So you notice your mites are coming in, you're getting high in July, early August. You can put this treatment on, and you've got a great option for bringing those mites down. This is a temperature-dependent product as well. Too high can kill bees, including damaging queens, and too low can reduce the efficacy. can affect the brood as well. So you will see some of the young eggs and sometimes larvae affected. Beekeepers are reporting anywhere from 75 to 95% efficacy. There have been other reports out there that are different than that. And some of that will be temperature and circumstance that have lowered the efficacy. So for the 14-day treatment, which is 2 pads at once, it does treat the mites under the cappings. So you're getting under those cappings and killing those mites. Mites seem to continue to come out of the brood, but they have been treated. And so if you're sampling, what we're finding is the mite levels seem to be elevated all the way up until 3 weeks after the treatment. And then magically, it seems that the mite levels start to fall after that 3-week treatment. So what I'm saying is don't sample once again until 21 days after you've applied the first pad. And that will be true, or the first pad, and that'll be true for the 20-day period. And I do realize you applied both at once in this one. So beekeepers report typically 80 to 95% mite kill. We've had some lower reports as well. However, once again, it depends on the circumstances. So this product is basically a single treatment where we are putting on the Formic Pro. So at day 0, products go on day 0. They don't go on day 1. Kills the mites under the cappings. So we have no mites re-entering. The 20-day treatment works a little differently. So this is 1 pad followed by another pad on day 10. And beekeepers are reporting that mites drop 4 to 5 days after the application of a single pad. This can be used with honey supers on the hives, but however, the difference between the 14-day and the 20-day is it does not seem to treat the mites under the cappings. And beekeepers are reporting 70 to 85% efficacy. So why are we seeing that difference between the 2 pads at once versus the 1 pad at a time? Basically, the difference is when we look at our mites emerging and mites entering new cells, we have a little gap in the middle of this 5-day period where they're not being treated by the first formic acid treatment, and they're entering the cells a little too quickly after before hitting the treatment 2 on day 10. So a few mites are slipping through and invading the new cells, and so we are seeing that it's not quite as effective. And then we have the same situation at the end. So 15-day period, basically we've got a few slipping through here again. Not a lot, but enough to reduce the efficacy over what we're seeing on the 14-day period. So some tips and tricks for using the Formic Pro. We have had some reported damage of queen, specifically with the 14-day treatment. Way to avoid this is to use a bit of extra ventilation or a little bit of extra space. Some beekeepers are putting in a box on or timing it so they still have a honey box on while they're putting on the treatment. It's absolutely the best option for treatment with supers on the hives at the moment. And mid-August, it seems to be the best time for both the efficacy and for the bee management and for beekeepers to do that. Now, this is for Saskatchewan. The rest of Canada, you're going to have to figure out what the best treatment period is for you. And once again, sample after 21 days after the first application. So when you put that pad on, that first pad, that's when you start your 21-day count. Other than that, if you do it before that, you're not going to see the full efficacy. So repeated formic acid treatments are another one that's available for use by beekeepers. And this one basically kills or treats mites a lot like the oxalic acid, and very similar oxalic acid cycle, and we're going to see in the next chart. And the difference is, instead of acting for about 3 days, we're seeing it act for about 1 day. And this one is only treating the phoretic mites. Once again, this is a lot like the oxalic acid chart, so I'm not really going to highlight to go too far into depth. However, you do see the mites emerging, getting treated on a 5-day cycle. It seems to be just enough in order to stop the mites from re-entering. So we can get some decent results using the repeated mite formic acid treatments. And then we have our thymol-based treatments, Apelifar and Thymovar. Once again, these are temperature dependent. They can suppress the brood somewhat. They really push the brood production away from the product, and they can also slow down feeding, particularly in the fall. They have to be applied twice for Thymovar or 3 times for Apelifar. And we're getting reports of up to 70 to 90% efficacy for Thymovar. However, we do have some beekeepers reporting significantly less efficacy than that. And once again, both of these products are very temperature dependent. And in Saskatchewan, our season is so short, and our application period and the timing for this product is so short. It's hard to get the full treatment in either in the spring or the fall, and avoid contaminating your honey crop. So this once again looks somewhat like the 2-treatment Formic Pro. We have the mites emerging. We put the treatment on at day 0, so treatment 1 goes on day 0, and the mites are killed. It's continually releasing. Put the treatment on at day 7. Product keeps releasing and killing the mites. I did this with Apelifar. If we did it with Thymovar, it's just 2 treatments instead of the 1 treatment. Thymol tips and tricks. We need to apply it as soon as the honey supers come off, which is late August. And you need to get this product on immediately in Saskatchewan. We don't have a lot of time before it gets cold. Sometimes as soon as mid-September comes, these products are no longer appropriate to use because our temperatures aren't warm enough. So get these products on in Saskatchewan as soon as possible. Your province, your region may be different. These products also seem to need time to start dropping mites. So seems like the thymol needs to build up in the mites' bodies in order for it to kill them. And so the mite drop will build over a course of a few days. Once again, get through your treatment and check to see if the treatment works after 21 days from starting. Oh, I have an extra slide in there, sorry. And then we have genetic resistance. So we've got these products. We know we've got Apelifar still working for the most part. It's got some issues. We've got these organic products that have issues. We need to combine other aspects. One of the more exciting ones right now is genetic resistance. We have breeding programs across the country. We have great breeding programs we can pull out of the States. And right now in Saskatchewan, we have higher degrees of resistance than we've ever seen, with better economics than we've ever seen, and 2 available queen lines that you can get in Saskatchewan are Saskatraz and Poline. Your provinces or states may have their own breeding systems or breeding genetics that you can pull the genetics from. We've actually just applied in the province for a strategic field program to demonstrate the ability of these selected stocks to influence the mite growth and compare the economics of these different stocks to the standard long-term Saskatchewan stock. We should have much more information to come about these stocks for future meetings. So plan to invite Medhat to come and talk about these in the future. So when we pull it all together, we're going to see a difference in how we have to treat moving forward. Historically, we've been using this blue line treatment where we put on a treatment of Apivar early in April, late in March, got a fabulous mite drop, taking it down to not quite zero, and then very, very, very slow build up through the summer, and then an increase back up to our economic threshold. And then we do a final treatment, which drops our mite back down so they're ready to be wintered. That's probably not going to be the situation going forward. We're going to have to treat once with an original product, be it one of these organics, be it Apivar. We're not going to get the same efficacy. We're going to see it climb back up to our treatment thresholds, and we're going to have possibly have another treatment early June, and then it's going to bring it back down, get us through our honey production season, and we have to start treating for mites through this period again. So we're looking at 1, 2, possibly 3 treatments. This is going to change. It's going to be different across the provinces, and this is a shout out to your tech teams and to your provincial programs to help you design programs for what you need to do. Remember, you're the beekeeper. You're in charge of your animal health and welfare. So it's you that's ultimately responsible, and the tech teams are there to help. So I'm going to bring this to a personal level. How am I pulling it together for me? What am I planning to implement? So this is, I've been trying to get ahead of where the beekeepers in the province of Saskatchewan have to be. I've removed Apivar from my program to treat the mites. However, I will still use it if I get in trouble. I will still be relying on the product to bring down mite levels quickly when I need to. So if Apivar is still working for you, by absolute all means, you can substitute something in here, somewhere in here. So my plan is to have 5 oxalic acid treatments as soon as the colonies are unwrapped. So we're looking at late April to start 5 oxalic acid treatments. Probably going to do a formic acid treatment during our early June dearth period. And I've chosen in this case to go with the Formic Pro. So I will be probably doing the 20-day Formic Pro treatment just to make sure my mites don't get out of hand going through the summer. And then I'm most likely going to use one of the 2 thymol products. I haven't decided which one yet, starting in mid to late August, as soon as the honey supers come off, and then oxalic acid to clean up. So fumigation of oxalic acid to clean up. By no means am I promoting any of these products. This is just one example of a program that I'm planning to use, and you have to design your own programs to fit your own beekeeping realities, your own seasonality, your own mite growths, and it's got to fit your systems. But the one thing I will say is this is significantly different than what we have relied on in the past, with a single Apivar treatment in the spring and maybe a cleanup in the fall. It's going to be more work, but to keep our bees healthy, we have to dedicate that work to our bees. And then I will leave that there.
Well, thank you very much, Geoff. I know it is especially challenging for you with the time difference doing this live from Thailand, but we really appreciate. We are a little bit late on the schedule, so I think I will just make a couple of questions, and then we will try to answer the rest of them, you know, in the near future. So we have a question from Michelle Clayton. Geoff, are there any theories or science as to why Alberta and Manitoba have much worse overwintering than Saskatchewan?
That is a tough question. Our beekeepers have been very, very proactive going out, and I can't really answer what's happening in the neighboring provinces. I can only really talk about what's happening in Saskatchewan, but I do know our beekeepers are very proactive when they call in at any time of the year, and I ask them what their mite levels are. They can say my mite levels are this. So they are very knowledgeable about what's going on in their hives. They're very aware of the health status of what's happening, and they're very proactive on what they're doing. So they're looking to see what the problems are. They're always evaluating if the products worked. They're always evaluating what they need to do next. And I can't really talk about Alberta and Manitoba without really delving into and having more information about what they're doing. But I think that's been key for the key to success is that dedication of our beekeepers, knowing what's happening in the hives and what they need to do next.
Okay, thank you. So next question. I think there are a couple of questions from me and Stepler that are maybe more focused on the dense transfer, so we can use those questions to make them after. Constantine, may we use oxalic acid and Apivar same time when Apivar is still in the hive? Also, may we combine different drugs at the same time, for example, Thymobar and oxalic acid, or Apivar and Thymobar, etc.?
In the province, we've had great success combining the oxalic acid with Apivar, and some success combining oxalic acid and Thymovar. I would not combine oxalic acid and formic acid. It's too much on the queens and on the hive. But we've showed that there has been enough safety for the bees to combine the Apivar with the oxalic acid and the thymol with the oxalic acid. Now you will see out there that there's a lot of comments that the Apivar and the oxalic acid may have an interaction where the oxalic acid breaks down the amitraz. However, when beekeepers have been using the combined, they seem to be getting good results controlling the mites. So it does seem to work.
Good. We have another question. Oh, well, the first question from Ian, and we can also open the question to other dense transfer team here. Do provincial apiculturists have data on the prevalence of amitride resistance within their provinces?
Some do, but we found it in Saskatchewan and we quit testing because we know it's here, and we decided instead of repeating to prove that we have a problem, we would address the problem. So our data ends in about 2021 or 2022. We decided we found the issue, now let's deal with it. Other provinces have continued to do the testing, and so they may have more results like that, and it may not be the provinces, it may be the tech teams, but there is some information out there, and a lot of them have it.
Do we have also another question from Ian that mentioned, do any of the provincial apiculturists test for amitride resistant mites when approving interprovincial colony trade?
No, that is a good question. However, it's extremely hard because basically we have requirements that mites must be under 1% for interprovincial trade. And to test for resistance, we need to have mites. So the mite levels are too low to test for resistance when we are doing interprovincial trade. So very good question. but we don't, it's very difficult to implement. So yeah, it's very tough. So kind of the theory there is if beekeepers are able to keep the mites below a level, they're successfully treating the mites.
Okay, thank you very much. We have another question from Kim Quebec. Is oxalic acid vapor heavier or lighter than air? Should we be fumigating from the bottom board or top of the brood nest?
That would be a great question for the tech teams, because I think some of them are doing the work. I have not done official testing. However, I have put oxalic acid in both the bottom and the top on my own personal hives, and it forms a cloud inside the hive up in both cases. So I've seen when I put it in the top, the oxalic acid will come out the bottom, and I've seen when I put it in the bottom, the oxalic acid comes out the top. So it can work both ways. Personally, I did not like putting it in the top because it seems to have more damaged bees from the hot vapor going into the hive than when you put it in the bottom. So it seems to affect the adult bees less when you put it in the bottom.
Good. I will make you the last question because I know it's late for you. It's more than 1 a.m. there in Thailand, and you probably deserve, some rest. So, Lynn Smith-Reeve, how many days after last treatment of oxalic acid vapor until we can put honey supers on?
That is going to change right away. The best option is going to be look at your label. So look at your label and look what the label says. So I suspect with the new registration, it's going to change. So make sure that you get the label and you just follow the label program. And I would like to address a question that was asked to the previous speaker about the tropolalaps combination and working together for tropolalaps, if that's possible. I'm actually here working with the UK research team on tropolalaps, and so there is quite a bit of international collaboration on tropolalaps work at the moment. So very happy to be here in Thailand working with Maggie Gill and her crew.
Well, Geoff, once again, thank you very much for connecting live, to do this webinar. I know you probably have faced a lot of difficulties in the last days, and I want to thank you, for all the efforts you have made. Thank you very much for the good presentation. I think I will just introduce our next speaker, which is Julie Ferland. Julie Ferland is Quebec's provincial apiarist. She's leading the bee health monitoring and biosecurity efforts at Medhat. She works with veterinarians, inspectors, and researchers to support the beekeeping industry.
My name is Julie Ferland, and I'm the provincial apiarist of the province of Quebec. I'm really glad today to present you the webinar about adapting Varroa management in commercial beekeeping operation of Quebec. The two main words to remember will be evolution and adaptation. And the take-home message that I want you to remember is that the old ways we use to treat Varroa mites will not work in the future. I wanted to start my presentation with a classic case of collapse because of Varroa mites that we receive each fall in Quebec. And each time beekeepers are convinced that Varroa is not in cause in the dead hives. So I received a call from a beekeeper in October. He has 7 to 8 years of experience. He managed more than 100 hives since the past 4 years. And at the end of September, the hives were looking good. They were taking their syrup well, but from the past week, half of the hives are dead. Several are dying. The remaining one are weeks and will not go through winter. They have no bees in it, and the remaining bees, there's a few bees on the bottom board, but nothing else. And each time beekeepers are convinced that it's poisoning because of pesticides. This kind of call, I always question the beekeepers about about his treatment practices, colony management, and IPM history for at least 2 seasons. In that case, in 2023, this beekeeper made no treatment in spring, only 1 application of formic acid during the summer, and only a few more formic acid application in September. In 2024, he made no treatment in the spring and summer, and only 3 application of formic acid at the end of September, which is really late for Quebec to start a treatment against Varroa. We usually recommend to start at the end of August. And as for the monitoring, this beekeeper did some alcohol wash, and at the end of June, he had an infestation rate of about 3%, but he was not sure. A veterinarian was sent the very next day and inspected the hives of 3 apiaries. Most of the hives that were still alive were weak and will not go through the winter. However, the nucleus colonies that were made during the summer were strong, and the bees collected from those nukes were collected and used as control for the analysis. The veterinarian collected the dead bees from the bottom board and made an alcohol wash with those bees, and the infestation rate was 8%. But that level was probably underestimated because it was done after 3 formic acid treatments. The bees that were still alive were collected for analysis and showed clear neurological signs. The result of the analysis showed no pesticides, both in control and sick bees. However, we detected the presence of 3 viruses, which are the deformed wing virus, the Israeli acute paralysis virus, and the Kashmir bee virus. The sick bees had a viral load much more higher than the control bees that were collected from nucleus colonies. So the cause of this collapse at the end of this season is really a combination of Varroa mites and viruses. The average mortality rate in Canada since 2007 is 28%. Each time there is resistance to the most used acaricide of the time, we see very high winter losses that are associated to that resistance. For example, in 2003 in Quebec, we had a 50% winter kill, and this is where we detected the resistance to fluvalinate. And the same thing happened in 2008 with the resistance of Coumaphos, which caused a high winter kill as well. For the most recent colony losses, we see more unusual patterns than before. In 2022, we recorded 45% losses both for Quebec and Canada. The following year, in 2023, we had 18% losses in Quebec, but the following year, in 2024, we had again high losses of 40%. And from all the surveys and visits to beekeepers, we were able to conclude that those high losses are really because of Varroa management practices, but not only because of Amitraz resistance. In Quebec, the situation is a little bit different from the situation of other provinces. The provincial apiarist, which is the work I do, is working under the health department of the Ministry of Agriculture, as honeybees are considered as farm animals in Quebec. The veterinarians take care of the honeybees along with other farm animals. And the Ministry of Agriculture offers various services to beekeepers. In Quebec, we have 1,500 registered beekeepers and about 65,000 colonies. Only 4% of the beekeepers owns more than 3/4 of the colonies in Quebec. We have about 50 beekeepers that are considered as commercial. In Quebec, the monitoring for Varroa mites is done mostly with sticky boards rather than alcohol wash. And as the treatment used, the most popular are organic acids, such as formic acid or oxalic acid, and thymol as well. And they are more popular than synthetic pesticides amongst beekeepers, especially in the last few years. In the past years, we surveyed beekeepers to try to see if there's a difference in the winter losses associated with the use of organic acid and or thymol only compared to the use of synthetic acaricides for at least once in the season. And for the past 10 years, we saw no significant difference between those two groups, as we can see in this graph. However, for the few beekeepers that use no treatment at all in their hives, we see high losses of 76%. So in Quebec, we suppose that if we had amitraz resistance already, we will see higher losses for beekeepers that use it, but it's not the case for now. So in Quebec, since synthetic acaricides are less used, it cannot explain all winter losses that we see in the last years. We then decided to revise all recommendations regarding IPN and best practices for Varroa management. We updated all the documentation and resources available for the beekeepers in collaboration with the CRESAD's tech transfer team in order to adapt Varroa management practices to these new challenges that we face. The key recommendation for Varroa management is to work with bee specialists from your province, such as veterinarians, agronomists, or entomologists. Then to monitor colonies on a regular basis and monitor a high number of colonies of the operation in order to better adapt treatment responses to Varroa levels, and also to include these new practices in regular beekeeping activities and then adapt them in the context of beekeeping seasons that start earlier, that are longer, and that are warmer than before. Then why working with several bee specialists with your operation? All of them have complementary experiences from seeing various types of beekeeping operation. Veterinarians have also experience of other animals, and this can be very useful for several issues related to honeybees. I really recommend you to work on a regular basis with a team of bee specialists. It allows them to develop their expertise as well as yours, and this will highly benefit the bee industry in regard to a bee health management practices. So don't ask them to come only when there's trouble in your beekeeping operation. Ask them to come on a regular basis so they know your operation when all is going well. So then when you have an issue, they will be able to help you more effectively. They will also be able to work on a global basis to enhance bees' health in general. For example, with a better nutrition and immunity for the bees, or to have a better rate of frame replacement, which will allow you to prevent bee diseases. Our recommendation is also to monitor colonies on a regular basis. On this graph, the gray area is the moment in the season when you should be monitoring for Varroa mites. It has to be continuous in the season to anticipate the buildup of mites, and not only at the beginning and at the end of the beekeeping season in a few colonies. You also need to monitor a high number of colonies. This will allow you to optimize Varroa treatments. In Quebec, we mainly use sticky boards for monitoring. This technique allows us to follow a high number of colonies on a continuous basis. Each beekeeping operation can see the Varroa buildup more easily and adapt the threshold for treatment for their own situation. We recommend to monitor at least once a month. The most important is to monitor before and after each treatment to assess the efficacy of treatment. It also allows to evaluate the remaining load of Varroa in the hive. For example, if your treatment worked well, but the initial load was very high, you will still have too much Varroa after the end of the treatment, and you will need to plan another one. You will not be able to see that without proper monitoring. We need to keep in mind that there is no one-size-fits-all formula for Varroa treatment. Each year, we need to make treatment decisions according to the current conditions and Varroa levels. We cannot continue to do what worked in the past because it's not working anymore. We can not only treat once in the fall as we did before, because now seasons are longer and warmer. We also need to treat in the spring to start the season with 0 Varroa. We then need to keep our levels low through the summer and treat when necessary, and finish with a fall treatment for the bees to winter with low levels of Varroa, and to start the following season with low levels as well. The key point here is to prevent damages before they happen, and allows the winter bees to be healthy for winter. Those winter bees are produced in late August and September, so Varroa treatment must be done before these winter bees are produced. The last recommendation is the integration of these new management practices to your regular activities. For example, you can develop the habit to check sticky boards each time you open a hive. It will take a few minutes more than your regular checkup, but will give you very important information for Varroa management. You can also adapt the timing of the treatment with conditions that occur naturally in the season. For example, when there's drought periods with no honey flow, or when you produce nucleus colony, when the queen stops laying for several days. These all can be opportunities for treatment or for monitoring. The actual recommendations are to do regular treatments with organic acids or thymol according to the monitoring results, rather than 2 applications of synthetic pesticides per season as it was done in the past. We do not recommend to use amitraz only, especially if you use it every year for several years. If you want to use amitraz, you must be very, very careful. It is very important to monitor before and after the treatment, and we also recommend to combine it with oxalic acid treatment at the end of the amitraz treatment. Now with the falls that are longer and warmer, we have more treatment options. For example, it is now warm enough for the thymol to be effective for both application of the treatment as per label recommendation. This was not possible before because of the cold weather in fall. And lastly, we do not recommend to apply several products at the same time in one hive. The monitoring on a regular basis and for each colony individually allows us to optimize treatment and avoid unnecessary treatments. This is important to not treat hives that do not require treatments because it allows the Varroa to develop resistance to those products. Each treatment has also a negative impact on honeybees, as a way to build up residues in the hive and cost a lot of money. The investment in time and monitoring will allow you to reduce the cost of unnecessary treatment. To facilitate decision-making about Varroa treatment, we created a decision tree for Varroa management. For every moment in the season, the beekeeper can decide which treatment to use according to Varroa levels, the presence of honey supers, the presence of brood, and the colony strength. The last tool we implemented to help our beekeepers are workshops. They were done in beekeeping operation with the collaboration of a veterinarian. We presented monitoring, best management practices, and treatment options. Since 2021, we did about 30 workshops. These workshops are free and allowed to educate about 400 beekeepers from everywhere in the province. They are very popular and appreciated by our beekeepers. Thank you so much for listening and for your interest in Varroa management. Don't hesitate to contact me if needed. Have a really good day.
Well, thank you very much, Julie. So I think it's time for the panel discussion. We will be discussing about Canadian bee health management with emphasis on Varroa treatments. So just to make, I would say, a quick reminder on the people that we will have together around. So we will have Maggie Lamothe. Maggie is a queen breeder and owner of Rayons de Miel in Quebec. She's a leader in Canadian beekeeping, advocating for sustainable apiculture and improved Varroa management. She also serves as second vice-president on the Canadian Honey Council and the first vice-president of the Quebec Beekeepers Association. So thank you, Maggie, for joining us. We also have Andrew Byers. Andrew leads the Atlantic Test Transfer Team for apiculture, supporting beekeeping and pollination in Atlantic Canada. His work includes research, education, and hands-on beekeeping. Thanks, Andrew, as well. Matthew Polinsky from Manitoba Test Transfer Program. Matthew is the program leader for Manitoba's Knowledge and Research Transfer Program. With more than 15 years in apiculture, he has worked in commercial and urban beekeeping worldwide and contributes to Canadian honeybee research. Thank you very much, Matthew. And last but not least, Medhat Nasr. Medhat leads Saskatchewan's bee tens adaptation program, focusing on bee pest management, breeding, and technology development. He has over 50 years of experience in apiculture research and regulation. So thank you very much all for being around the table to answer all the questions we have been receiving. I have plenty of them. So I will start with the first one. Maybe you can just make a very quick, I will say, view, share your thoughts on what are the most serious issue the industry is facing in the different areas that you are managing. So maybe we can start, for example, with Matthew, just a few words presenting what's going on in Manitoba.
Yeah, sure. Thank you. In Manitoba, I guess, well, first I just would like to mention briefly some of the presentations we had, which brings me to what's going on in Manitoba. Geoff emphasized our treatment regimes are shifting, as maybe we haven't been seeing the same early season knockdown that we have historically, and we need more attention and labor for that control. And Julie's presentation demonstrated exactly that, where in the case presented to us, the beekeeper in question had skipped multiple treatment windows, which may have been more common in the past, but now, and especially in light of potentially shifting thresholds, is less of an option. So now our position as beekeepers is not only capitalizing, on the treatment windows, but basically prying open that window and trying to fit them in where we can based on our environmental conditions and hive statuses. And specifically in the fall period, as soon as the honey comes off, and perhaps foregoing that extra 5 or 10 pounds and making sure we treat in a timely manner for our respective regions, I think is critical. And again, that just brings me back to what's going on in Manitoba and brings me to the point of monitoring levels. So just as we've heard throughout today, we're looking for standardized methods of monitoring, consistency in monitoring, record keeping, and I hope we'll have a chance to talk a bit more about that on how to get an accurate picture of mite levels in your apiaries and just making sure we're on top of it. Thank you.
Good. Thank you very much, Maggie. I think you can also share your view from Quebec.
I think it's actually like two main problems I foresee. Illnesses, which are linked to the mite, but are also linked to the environment through, for example, contaminants and pesticides. There are a few articles that have come out, research that has proved that the presence of pesticides in the environment equals an uptick in the presence of mites, or the mite parasites, the quantity of mites in the hives. So that's quite problematic because we do not, as beekeepers, always have an impact or a control on the bees' environment. And there's also access to queens. Actually, I think it's a major issue in Canada, especially in springtime, where we don't have access for many reasons to local queens. So I think those are both very huge problems we face.
Thanks a lot, Maggie. Andrew, what about the Atlantic Canada?
Thank you, Miguel. I'd just like to also express that this has been a great afternoon, and we're all up against the same challenges. And I know, being a small part of the group that organized this, I don't give myself much credit but for the people that did the heavy lifting to pull this together, we all need to be really grateful. Hopefully the start of something that we'll see in our beekeeping calendar in the years to come, talk about important bee health issues. I think in the Atlantic region here, we have to talk about our Atlantic region a little bit differently, because as I said before, we have one province that doesn't have Varroa mite, and we've got our fingers crossed that that's going to be the situation into the future for Newfoundland. But when we look at the Maritimes, we got a wake-up call last year. Mostly when you pool the maritime provinces together, our winter losses as kind of an assessment of how well we're doing as beekeepers and managing our Varroa are very good. Nova Scotia, for example, leads the country, aside from Newfoundland, which isn't a fair comparison, but Nova Scotia leads the country usually in winter losses. New Brunswick's usually within a couple of percentage points, and then PEI does have some variability in winter losses. But last year we saw those, what we're expecting normal winter losses or acceptable winter loss between 15 and 18%. That's usually where we fall in Nova Scotia and Brunswick, and they jumped right up to 30%. So that was kind of a shock. We're watching things really carefully this year to see where we are, but we feel that, and with not only talking to beekeepers, but the work that we do with ADA, that Apivar is still mostly effective. We're very cautious saying that, and we're very cautious in how we're using Apivar for a lot of the reasons that were described today. But we typically are seeing that Apivar is being used by most of our beekeepers, particularly commercial beekeepers, once a year, with then some other treatment, typically again an organic acid. So that seems to have been working for us, but we are monitoring that both to get real good, credible results through the work of ADA and also talking to our beekeepers. But a couple of other things that we're seeing that are of concern to us here, we're seeing winter losses that are hugely variable. We're seeing beekeepers with up to 100% winter losses in some cases. And then we're seeing beekeepers that are managing their mites and keeping their winter losses within that, 15 to 20 or 15%, 18% range which is what we want. So that's one of the questions we have. Why are we seeing now this emerging real extreme variability? And also when we're going into yards to monitor for Varroa, what we're starting to see is that a yard, for the most part, will have reasonable Varroa, what we want to see, very low, manageable populations of Varroa mites. And then within those yards, individual colonies that spike and have high Varroa. So these are some of the things that we're seeing. They're challenges for us. We don't know what the answers are yet, but we're looking at it very carefully. And also the mid-season treatment is an area that we're starting to look at. We're extending our seasons now with climate change, and we really have to concentrate now on coming up with good answers, good solutions for a mid-season treatment. So those are some of the things that we're kind of concerned about and looking at here in Eastern Canada.
Thank you, Andrew. I think we're doing something right, because after almost 3 hours of webinar, we still have more than 200 people connected. So for sure, there was an interest on the speakers and of course on the panelists. So Medhat, I don't know if you want to also share your view about what's going on in Saskatchewan.
First of all, I'd like to thank you personally, because you started when you called me up and said what we can do to help the Canadian beekeepers. And the whole idea came about this workshop and webinar. In Saskatchewan, actually, it's still number one, according to our reports from the beekeepers. Varroa mites is number one challenge, how to keep it under control, especially under our prairie conditions. Our season is quite short. Then we have our honey crop. And just between taking the last honey supers off and set of winter and cold weather conditions, it's quite short time. So the beekeepers really have to be right on the top of applying the treatments when it is suitable to have effective results. And the challenge now, some beekeepers still like to have the last honey drop and take it off the supers and still believe they have enough time to control the mites. However, that short period of time, it is not really enough to get the mites under control. So that will require monitoring the mite through the year, have very effective treatment in the spring, and that is to keep the mite population down until they get into the fall. I don't like that when we talk about fall as a season, it can be 1 month, 2 months, 3 months, but our treatment, it should be based on our mite level and having enough time to allow our treatment to work to keep those bees healthy going through the winter. So that's a mini challenge we still have. And some beekeepers actually very successful in organizing their work plan, and they can bring it down to about 10 to 15% or less than that mite or colony kill. So the key, treatment at the right time, following the monitoring to make decision. Thank you.
Yeah, thank you, Medhat. Yeah, we are really glad, that our first idea of supporting Canadian beekeepers has converted into a series of webinars, being supported by Canadian Honey Council and the Bee Transfer Program. So thank you all for being here and for making this possible. I think we heard a little bit about the IPM today. What do you think? Are beekeepers successfully able to implement this strategy? What are the problems in implementing this strategy? What are your thoughts about that? I don't know, Matthew,
Miguel, who
you want to
to
answer,
start?
Andrew? Yeah, go ahead.
Yeah, no, happy to comment briefly, and then I'll let the others pitch in because I know our time is limited. We've always felt that the reason we have success here in Eastern Canada, as I've explained, is because we have used aspects of integrated pest management in managing Varroa. We haven't relied entirely on one single chemical treatment. We've used chemical treatments, we used organic acids. Monitoring is something that we've always encouraged beekeepers to do, and I think that vigilance with monitoring is increasing. So I think all those aspects of integrated pest management or integrated resistant management have kind of fallen in place. So, we're cautious as we move forward. We don't know what the future holds, but we're hoping with that approach, which we've had for a number of years here, we'll continue to manage our mites successfully.
Yeah, I think we are all waiting for new actives, to be available. But in the meantime, of course, I think the best strategy is to be cautious and to use the tools we have already available in the toolbox. So Matthew, I don't know if you want to add something, or Medhat, feel free, or Maggie, sorry. So
I'd like to hear from Maggie, her system for managing the mites in her operation, and that will be great if she can share with us what she's doing in terms of monitoring and treatments.
No problem, Medhat. I'll do a first little talk in English, and then I can translate for the people online. So on the farm here, I have one golden rule for every beekeeper that comes and works with me. And that golden rule is mites, 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 monitor the mites. So every time we go out to the yards to actually work on the bees, the first thing every beekeeper will do is actually take out the tray, because we work on the farm with AP Novar screen bottom bards, which I hope we will be able to produce here on the farm shortly and maybe sell. But so we'll take out that tray and count the mites on that tray. Even though it's been there for 10 days, I don't care. We count them, and we'll keep track of this on our, we have an online application with AP Tracer, but we also write it on the roof of the hive. So we will have a count. For example, let's say it's 5 mites with the day of that counting. So if it's, for example, the 10th of June, and then the following time we come back to that yard, we'll get to that same hive and recount a second time, because we go to the yards probably every 10 days. we'll take out the tray again before working the hive and count those mites again, and then we'll write it again on the roof. And then we have those 2 dates and the quantity of mites counted each time, and then we can divide that quantity by the number of days. Yes, sometimes we have a harder time counting the mites because there may be a little bit more of wax or debris in the bottom board, but by doing these frequent counts, I'm able to get probably around 15, 20 mite counts per hive, and I do that on my 700 hives every time we go through them. So I will get a lot of data from these mite countings, and I can spot every mite bomb in the yards and do specific treatments on these hives. So I save a lot of money on treatments, a lot of time on going around to yards for nothing by just knowing there's no mites in every yard, or in these yards I don't have to go and trade because there are no mites. I just go to these specific problematic mite yards or bee yards. I've noticed through the years that the yards that usually get more infested are the ones that are closer to towns. I don't know why. Maybe there's a bit more of small beekeepers, so maybe there's a little bit of a harder mite management over there. I don't know. This monitoring gives me the ability to be proactive, exactly what Geoff said. So I really want to put emphasis on that proactivity. You have to see the problem before it arrives, and that's what this monitoring on the farm gives me a chance to do. And that gives me a chance to time actually the treatments. So I know the problem is coming up, and I know that I can take off the honey boxes, or I know when I'm gonna do a first honey pull, and then I can go in quickly and do that treatment. And it also gives me a chance because I do count the mite drop after treatment because we go back in the yard. So if I use, for example, multiple oxalic treatments, well, I'll do a counting every time. So I see my mite drops, and then I know I don't have any more mites in the trays after, for example, 4 treatments of formic acid, because there's not going to be any more mite fall. I will probably do a treatment or another treatment just to make sure, but I really make sure I do the proactivity Geoff talked about.
Thank you, Maggie. Well, I think we can keep going with another question that we have received, for example, which is related to some of the speech of the ideas that you included. What are the proper economic threshold to use for mite control? How mite treatment threshold can be calculated in different parts of the world? You know, people considering different genetics, different type of bees, different climate conditions. Is there some specific or some scientific paper or protocol addressing this topic?
I can take this question. Actually, if we look at the literature, because I'm doing this work right now, we have only 2 papers discussed and the economic threshold development, and both of them since 1980s. One by Keith Delaplane from Georgia, University of Georgia, and the other one, it was done by Rob Carey from University of Manitoba. Other than that, the economic threshold based on those ones kept changing based on experiences and reports by the beekeepers, and working with the regulators and looking at what is the proper threshold. In the Bee Health Coalition, they have a document, it's called Tools for Varroa Management, and there was proposed threshold out there. And as a matter of fact, right now we are into the 9th edition of that document, and we have about 10 reviewers. It was first developed by Dr. Dewey Karen, and we are 10 reviewers working with him to update the information. At this time, we are saying it has to go with the bee population phase, and that really to consider the regional differences in terms of mites, in terms of bees development. For example, up in Canada here, we have long winter, short season, so that way we can relate the threshold to the bee development in Canada. If you are in southern states, you have almost 10 months during the whole year, and the bee population, it change in terms of the dynamics and development in that area. So we have actually for the honeybees, a growing phase, which we call it in the spring. Then we have the summer, and that's during the honey flow. And later on, we get into the fall, which the bee population will go down. And later on, we have the short period or long period based on where you are at winter time. So for the population increase, like in the spring, we are looking at mites less than 1%. Right now in the recommendation in that document, 2%, but we are in discussion to lower it down to 1% to make sure once we bring the mite at this time before the honey flow, less than 1%, you don't have to worry about the mite peak during the honey flow and you panic. In terms of the fall, and here it can be major difference, we still looking at once you see 1% of mite level in August, early August, that will give you warning. You have to start considering treating as soon as you can. 2%, that basically says treat today. So we are looking at 1% now in the spring to trigger the treatment. We are looking at 1% in August. So that will give us time for us in Canadian climate to consider the treatment and killing the mites before they can inject their virus into the winter bees. So that way we can keep healthy bees. So in short term, 1% in the spring, making sure going to the honey flow with less than 1% and 1% in August or mid-August. And that's to make sure our treatment is going to work to protect winter bees. And my colleague can add any addition here if they would like to put some inputs here.
Can
I'm happy
I say? Oh,
Yeah.
sorry.
No, you go ahead, Maggie. There's a slight delay which makes us talk over each other, but you go ahead.
I can say what my thresholds are on my farm as of today. I do spring treatment, so my threshold in spring is 0 mites in the bottom board. That's the aim. So we give a go. We will go like 5 flashes of formic acid or 5 oxalic every 5 days, like Geoff explained. And then mid-season, if I see, if we arrive in July and I start seeing, and that's on a 10-day basis. So if I start counting 5 mites for 10 days, which is about like 0.5%, I think. If I see 5 mites, we start planning treatment on that hive or on the bee yard, depending on how many hives I detect having this many mites on the bottom board. I start planning what my next action is. In autumn, I again aim to get back at 0 mites in the bottom board after treatment, ideally, or might fall for more or less 10 days. So that's about my thresholds on the farm.
That's using sticky boards. You don't use the
Yes.
shaker.
No,
You
I don't
use-
use the shaker.
Yes.
I don't.
Okay.
No, that's not true. I use the shaker because I work with the research center in Quebec on the apiomics that genomic selection we're doing on the queens. So I do have shaker results, but not on all the hives, on specific hives for the selection program. But I prefer the bottom board because I find the shaker is kind of random. Because you kind of get 300 random bees. Yes, they're from the brood box, but you have to be lucky to get the bee that has the mite on its back in the brood chamber. And Geoff was explaining that like less than 25% of the mites are on a bee back, so they're phoretic. So I have a very large preference for the bottom board because the mite has no choice but to fall on the bottom board when it dies. So I find it a lot less, not sham, but a lot less left to chance, if I may say so.
Andrew?
Yeah, well, in the meantime, a couple of the points I wanted to make were raised, which is great because we're all presenting the same message. The situation's changed, and when we're looking at monitoring mites, it's really important to understand a couple of things, and Maggie's alluded to them, but maybe I can just expand on that or explain how I kind of think of it. Because if you're looking at sampling with the alcohol wash, as we call it, you take approximately 300 bees from a colony of say 30,000 plus, and that's your sample. But if you're using the excellent system that Maggie does, where she's monitoring the entire colony, your sample goes from being 300 of 30,000 to the entire 30,000. So you're getting a much more accurate result. And when people are looking at alcohol washes, it's great. And it's far, far better than doing nothing. You have to be monitoring. But you have to also understand in some of the research that we do, and Medhat and I have talked about this because he's doing something similar at the moment. We have to do 2 washes on the same sample, just as part of the methodology for the research. And probably about 20% of the samples, we find additional mites on our second wash. So this comes back to what Maggie's saying is, it's certainly good to do the alcohol wash, but the sample size is tiny, and the chance, if we're looking at the presence of mites, which is what we're actually talking about, we used to talk about 3%. Now we're not. We're talking about 1%, and essentially 1% is if you're seeing mites in your alcohol wash, for the most part, you're getting near or reaching that 1%. 3 mites in a 300 bee sample is 1%. So we're looking at a very small number of mites in our sample. So because that's where we're going now to make considerations or determinations about treatment, we have to be very careful because it's easy to miss that 1 or 2 mites that we need for that sample. And we're telling beekeepers now, and again, this aligns exactly with what Maggie and Medhat are saying, when you're seeing mites is when you need to start thinking about treatment, not when you're getting 3%. When you see a mite, that's when you need to intervene. And by monitoring carefully and being vigilant, as I've said, and understanding your own bees and the patterns of Varroa population growth in your colonies, then you will make decisions about, do I need to step in? But certainly when you see a mite, that's when you need to probably treat, but at least start considering how am I going to manage this? Because if I wait until I get to the 3%, it will be too late. So that's the situation we're in now. If there's presence of mites in your sampling, you need to consider treating, which I think is the same message we're all trying to convey.
Just to build off that a little bit, we see that with Maggie's strategy, she's monitoring many colonies. When I get calls from beekeepers and they're talking to me about their mite levels, I always ask how they monitored. And one thing I've been noticing in this past year, for example, is with the alcohol wash, people might monitor 2 colonies or 3 colonies or 8 colonies per yard. And then it's also they're collecting different numbers of bees at times. And then we have beekeepers who are doing pooled sampling, and then they'll collect 200 bees per hive or 100 bees per hive or 50 bees per hive. So it's been really difficult difficult and also important to figure out what people are doing, and then make sure that everyone is on the same page doing the most standardized testing. I mean, Maggie's is incredibly standardized, and with the alcohol wash, we all know that the standard is 300 bees per colony. And Andrew was kind of emphasizing, even with that, you're missing mites at times, and Maggie emphasized the randomness of it. So I think more conversations on monitoring and some of the research that Medhat's doing on understanding how to detect mites is super important.
Medhat.
David Karen is here joining us. If he can join us here and give us his opinion.
I'm Dewey Karen. I am the principal author of the Honeybee Health Coalition Tools for Varroa Management. I worked in bees and pollination on the East Coast, and then since 2009 have been on the West Coast in Oregon, where I continue to work with our Master Beekeeper program.
Okay. Go ahead. If you want to add something here, you have your hand raised.
The Honeybee Health Coalition Tools for Varroa Management includes both the recommendations for the U.S. and also for Canada. It's an IPM approach. We do have the specific information on the various chemicals. We are in the process of revising it now for the 9th edition because of the additions of some new compounds, and also for this very same thing that has been talked about across the various Canadian provinces, and that the old way of doing things, which has been, we use Apivar a couple times a year, is not working for our U.S. beekeepers as well. And so they need to integrate other methods into their management. And as you've heard from the various provinces, there's no one way for every beekeeper to have this work for them, but it does involve this integration, also for resistance, plus also to get the efficacy that they expect. Our recommendations have been coming down over the years as we've revised this, and we too are at the point to say 1%. If you see mites, particularly in the spring and spring monitoring for mites, it's not sufficient to conclude that, well, my colonies don't have any number of mites, so I don't need to do some treatment. Mite control needs to be, I think is very well illustrated, something now that is season long and an integration of methods to try to control. Our beekeepers, just as you have in Canada, have a real windows in which they can do their treatment. Our beekeepers are moving colonies a great deal more than those are moved in Canada for pollination purposes. So they have windows when most of the colonies would be in one place. When the colonies are scattered across several different pollination locations, it may not offer an opportunity to do the treatment. So part of our additional issue for our U.S. beekeepers is having that appropriate window in which they can apply these variety of controls.
Thank you very much, Dewey. So I think we can follow up with another question that we have received in the chat from Dion Bacon. How soon will we see research results from the various indoor treatments being tested for those that overwinter indoors?
As you know, we have had a very heavy loss of colonies reported by our commercial beekeepers. 700, almost 800 now have been surveyed by the group called Project Apis M. Losses were very extensive, above 50%. The individuals that put colonies into indoor wintering additionally as well had those level of losses. And the comments mostly were that, while the colonies going into the houses were not as robust as they have been in some past years. And so the winter housing is not a remedy. If the colonies are not strong enough going into the houses, they are coming out in that same weakened condition. And the level of losses of those doing indoor wintering was not different from those that were doing outdoor wintering.
The only early work was done by Rob Carey about using formic acid fumigation indoors, but that's really not commonly adopted by beekeepers. And for treatment indoors, we just started initiating some research in Saskatchewan with the College of Veterinary School to look at using maybe the oxalic acid in glycerin application indoors, but that's really, the research is about to commence this year, and we don't have a really effective way of applying the products indoors. Again, the risk will be too high for the applicator, and we have confined bees indoors. How that will really impact the bees during the winter time, we don't have results or data to support any recommendation at this time.
That would be the same for the US. There is a little bit of work going on. The problem is that the colonies are so packed into the structure that it would be very difficult to individually try to treat colonies. And it's a far reach to think that we could do a whole house treatment that would ever be effective.
Yeah.
We have another question from John Murray in the chat. He mentioned that with Varroa causing significantly more challenges, and for example, with the strategy proposed by Geoff during his presentation, do you think that with all these treatments, we will see a reduce in the virus pressures at the colony level, or how's your view on that?
Still here. Always Geoff, he has that idea. If you don't have Varroa, you don't have virus. So that's basically when somebody asks him about virus, he say, forget about it, just deal with Varroa. And I think he's rightly so, because Varroa is a vector for viruses. And once we have higher level of Varroa, we expect higher level of viruses. So that's why we have to be really paying most of our attention and focus on killing the Varroa at the right time before they vector the virus to the bees.
One additional concern we have in the U.S. is that it seems that the DWV virus type A is evolving towards a much higher prevalence of type B. And type B seems more virulent, and that may be one of the reasons why we think we're getting the failure of the Aprovar treatment, because we have a virus that's more virulent.
I can add that you have to be very wary when you have a bad mite year, so a high hive loss, because the viruses will be, or the illnesses will be more present in the hives in general, and you will have maybe a few years, probably a 2-year remission of that loss. So the first year you'll recoup probably the quantity of hives, but that does not mean all the viruses will be gone from your all of your beehives, because as we all know, a generation of sick bees will have a hard time to cater or care probably for the next generation of bees, and so on. So it will take a few years to of diligent treatment and no mites and care that you will have to give back to your bees to be able to kind of get that mite or that virus load down, or that illness load down from your whole apiaries and bee yards.
Okay,
Maggie,
I think we
this
are.
is Dewey again. Maggie is right on, because our beekeepers last year did have heavy losses. And although the reports are for this year, this is their second year of the very heavy losses. So Maggie's right on with her comment.
Okay, so I think let's try to make a couple of last questions. I think we are running out of time if we want to stick on the plan. So we have Eduardo Gomez. In my experience, the vehicles we use to deliver the acaricide are just as important, if not more, so than the active ingredient use. What new vehicles are being evaluated to apply the current active ingredients?
In terms of the vehicles being used to deliver current active ingredients, I think Véto-pharma has been mixing it up a little bit with Amatraz. I'm not sure. Maybe you can speak to that. I noticed that there's one being used in New Zealand. I'm not sure if anyone here has knowledge on that, calls a Nasr for evaporation of formic acid. And of course, in Canada, we're working really hard on delivering oxalic acid through the glycerin method. A lot of the tech programs have done work on looking at the efficacy and safety of it, and now it's just getting it registered. So those are a few that come to my mind. If anyone else has comments on other vehicles for delivering the current active ingredients.
No, I think you're right, Matthew. At the end, what is important here to highlight is that, of course, it's not only a question of the active; it's how the formulation, how the active is being available, and it's not only a question of the work of the active itself. So I think it is very important the bioavailability of the active, you know, to make I would say the final release on the product. Anyway. Well, let's pick on. Well, we have one question from Marta. Marta, just regard from here with Quebec having lower use of synthetic acaricide. So I think that's for Maggie. It makes sense to think that resistance is less of a challenge. However, is there data confirming the lack of resistance, and do all synthetic miticides always work when applied?
I cannot answer to the presence of resistance. I think there has been a start of it, but I don't have data with me to be able to share that with you. Julie Ferland might have been able to give you guys a bit more info on this. For me, up to now, all organic acids have been very good, and I have not seen any presence of resistance to these treatments in my hives. They've been very, very good. We've had a presentation from Benjamin from Epinov in France the last three weeks ago, I think, and he was talking about the risk we are taking by using glycerin and oxalic acid during long periods. He's wary that we might accelerate or create resistance of the mite to oxalic acid because of the length of time the product will stay in the hives. So that's kind of the thought for the future. We have to be very careful. He was talking about the fact we use the flashes or the short-term applications, they're stronger. So the risk of creating resistance is probably lower than lengthy, lower dose treatments. So I don't know if that answers the question.
Thank you, Maggie. Any other comment, Medhat, Matthew, Andrew? Do you want to add something or?
No, but also like we are looking at, because we are using heavily here oxalic acid sublimation or vaporization. If somebody using Abivar and might survive that treatment, we know those mites are already resistant. So in reality, when we come and treat those hives with oxalic acid, we are killing those resistant mites. So if there's any other, that's part of our management of resistance by killing whatever residual might survive the application from Abivar. And that way we can reduce that genetics development inside the hive with the other mites coming back the following year. So that really help us now to extend the longevity of using Epivar as part of our treatment. So that variable organic acids used in the hives, and that's what is common in Quebec, that's helping really to reduce the rate of developing resistance. But the results, as Maggie said, Julie will have more information about that.
Yeah, just speaking
Matthew.
to Martha's question as well as that earlier one from Ian about Amitraz resistant documentation across Canada. Yeah, tech teams are doing work. Sometimes the government, the provincial aparists are doing work. I know that people want to see more data. There is some data being published and it takes time, but I hope that we can continue working together across Canada with standardized methods, doing both the field bioassays and the genetic genotyping to be able to have more of that public and consistent. I think it was referenced earlier by Geoff. You need mites to be able to test, and it takes a long time to be able to get those results in place. But I hope that's something we can all work on together and discuss further in the future.
No, Miguel, we're running out of time, so I'll just add to that we're all doing work on this in one form or another here in Atlantic Canada. The tech transfer team is working on looking at efficacy of Epivar. I would urge beekeepers, if they want to know what's going on in their region, to reach out to their tech transfer team and their departments of agriculture, attend upcoming meetings and workshops, because at those we talk more fully about the work that we're doing as it applies to our region in much greater detail than we have the time to talk about it today. So that's what I would say to beekeepers. If they want to know what's going on with miticide efficacy in their region, get involved in those activities.
Good. Thank you very much. So I think it's time to start closing the first webinar as we are running out of time. So I would like really to thank all the speakers. We have Phil Lester this morning starting at 5 a.m. his time. We have Geoff connecting at midnight in Thailand. All the people across the borders. Thank you very much for the panelists. Thank you very much, Matthew, Medhat, Maggie, Andrew, for having the time to spend some time with us. Thank you very much to the Canadian Honey Council and the Canadian Bee Transfer Program as well. And of course, all the attendees of the event that are survivors, after 3 hours and 40 minutes, still being connecting more than 200 people. So thank you very much all for making this first webinar a success. Just like a small reminder, we will be sending in the coming days an email with the access to the online presentations, which we hope to have available in both English and French. And as a reminder as well, this is just the first webinar of a long series that we hope to continue next fall with new and exciting topics and, of course, new speakers. So wishing you a wonderful Saturday evening or afternoon, depending on where you are. And thank you very much. See you soon. And thanks for all the support. Thank you. Bye-bye.
Bye-bye.